So you got
Oh,
out just in time.
oh, still locked down there, right?
Yeah,
Oh,
so Auckland is kind of mostly locked down. I think takeaway restaurants are open, but that's it. And then they opened the high schools to the final 3 years of students,
okay.
maybe about 2 weeks ago.
Okay.
And that's about a third of New Zealand is in lockdown. And then I'm just to the south in Hamilton. Hold on, I'm trying to get the
Is the university
sure.
closed or?
So the university has been closed since
Ah.
August 15th.
Oh,
Yes. So
okay.
always fun. Okay, let me see if I can.
Can we start, Briwayan or?
Yunus, please.
All right.
How's that?
Yes, it's better. Caroline. Well, anyway, thank you very much, Bliwayan, and good morning, everyone. Welcome to day 4 of the Mangrove Monitoring International Training 2021. And today, Thursday, November 4th, 2021, we have a special session with our invited speaker, Dr. Carolyne Lundquist from the University of Auckland, New Zealand. And she's going to explain about the topic, integrating socio-economic, political, and scientific factors, strategies for mangrove management. And this session will be moderated by Dr. Suyadi from the Research Center for Biology Brain. And Dr. Suyadi, time is yours.
Okay, thank you, Mas Yunus. Good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. Today we have in the 4th day for the Mangrove Monitoring International Trainings, we have excellent speakers, Dr. Carolyne, and she is already here. And then before she start her speech, I'm going to introduce about the Dr. Carolyne. Carolyne Lundquist moved to New Zealand in 2000 after obtaining her PhD in ecology at the University of California, and she also did a PhD in marine biology from UCLA, and she holds joint position as a principal scientist in marine ecology in the National Institute of Water and Atmospheric Research, or NIWA, in Hamilton, and she also as an associate professor at the School of Environment at the University of Auckland. Actually, she was my PhD supervisor in New Zealand, and her research interest is very diverse. It's ranging from scientific research to inform the management of mangrove and other coastal wetland habitats, to reviewing the impact of climate change as well on the seafood sectors, and development of marine spatial planning tools to improve management of accumulative impact in New Zealand's ocean ecosystems, to the development of global biodiversity as well. She has served as a co-chair of the IPBES task force on the scenario of models of biodiversity and ecosystem services experts group in 2016. The task force has been developing in a new global biodiversity scenario framework entitled the Nature Future Framework that is currently under the review of IPBES, governments and stakeholders. And she also have a long experience research in mangrove, especially temperate mangrove is quite different from tropical mangrove, especially in Indonesia. So this will be very interesting and will provide new knowledge or experience. So welcome, Dr. Carolyne, and times and use. We have maybe plenty time, so you can explain and you can share your experience to all of us. Thank you.
Great, thank you, Suyadi. And am I sharing my PowerPoint from here?
Yep.
Okay, let me get that to open up. Ooh, security alert. Oh no. Hopefully that's okay. There we go. So thank you very much for inviting me to participate in this training course. I know I'm here in New Zealand, but I'm originally from the United States, though like most of the world, I haven't been able to travel for a few years. So I've been 2 years now without seeing my family, which is quite sad, other than my husband and children are here in New Zealand, and they've had, I think, maybe 3 months. Most of the last 3 months, my daughter has not been in school, and my son just started school back up about a week ago. So I think we're all in a very similar, very strange world the last couple of years, but hopefully the world is moving out of that, where you'll find some things are, I think New Zealand has a reputation of being quite different from the rest of the world in things that it does. And I think that is the same with mangroves. So mangroves in New Zealand were temperate mangroves, and so temperate mangroves are about 2% of the mangroves in the world. And I think the way people treat mangroves in New Zealand is also quite different. So what I thought I would do today is go through a little story of what we know about mangroves in New Zealand, but also a bit about their expansion. So how many more mangroves we have every year. And then a lot of that then becomes a very social issue in New Zealand. So in many places in the tropics, the challenges might be on different uses of the coast where people want to build infrastructure or otherwise impact on mangrove habitats. In New Zealand, we actually have mangroves growing in places where they weren't before. And so people often see them as a pest species and want to remove them. So it's a very, very different dynamic that we've moved through. So I'll give you kind of a story of the last 50 years or so of mangroves in New Zealand and showcase how it's both important to understand the science as well as the social science and how communities and indigenous communities work with mangrove ecosystems as well. And of course, I've got a whole ton of pictures, and Suyati might know some of these people are various students and staff. So Rich Bulmer here on the left squatting in the mangroves was a PhD student about the same time as Suyati, and I think Rebecca Gladstone Gallagher in here as well as another PhD student. And then there's a whole bunch of other students, and also, of course, I always put my children up here. So my children I drag out in the field with me as they've been growing up, and they're now 13 and 15. So they don't like to go out so much with me anymore. So what do we know about mangroves in New Zealand? Well, the first thing is we've only got one species. So we have Avicennia marina. So that's one of the very common species around the world, but particularly in temperate latitudes. And they occur, I think I've got another picture that'll show you all of New Zealand, possibly. No, just the North Island on the next slide. But this is just kind of the upper chunk of the North Island of New Zealand that shows up here. This and Auckland is right here. I don't know if you can hopefully see my pointer, but we've got Auckland right here in the Coromandel Peninsula, and going down to right about here on the east and the west coast at about 38 degrees latitude is our southernmost limit of mangroves in New Zealand. And they exist as they do everywhere, just in that intertidal zone between mean sea level rise and that highest astronomical tide. And we know that they're going to move with sea level rise. So we know there are depths that they aren't able to survive at if they're inundated too much. But we also know that we have a big problem in New Zealand because often what we have on the other side here of on the coastal side is we usually have infrastructure and houses and agriculture. So often we don't actually have anywhere for mangroves to go once the sea level rise takes out this part of the habitat as it becomes too deep, that they will actually have challenges in moving shoreward. So that's kind of one of those great ecological challenges that we have in the near future. On the other hand, we do have a lot of sediment inputs. So as we're looking at changes in sediment, and I'll showcase some of Suyati's work, where we have sediment coming in, we're actually creating more seaward habitat for mangroves. So depending on which of our climate change scenarios, it's unsure whether we're going to have more or less mangroves in the future in New Zealand. But the key thing that's very different about mangroves in New Zealand compared to the rest of the world is that in New Zealand, for the last about 70 years, we've been averaging an increase of 3 to 4% per year increase in distribution of mangroves. So this is ongoing, and I'll show you a few of the reasons for this as we go through the talk. But first, there's that challenge in mangroves. So people either love them or they hate them. And there have been a whole series of articles about mangroves. We've even had a mangrove bill. This is to allow all removals to occur without any requirements for permission, and that went to a parliamentary select committee. So we actually had New Zealand Parliament meeting to discuss whether or not it was okay to remove mangroves. So with 3 or 4% increase in mangroves, a lot of people don't like them because they see them as being associated with muddy and smelly sediments. They see them as blocking their access to the coast. And so this has been actually quite interesting to watch as this has changed over time from about the 1980s, and I think that's my next slide, all the way through now, where in the 1980s, we had basically global mangrove conservation movements really having an influence in New Zealand. So most of the papers and the research in New Zealand in the 1980s was about what do we know about mangroves and how do we protect them. But then by the 1990s and 2000s, suddenly people were starting to notice that mangroves were in a lot of places where they hadn't been before. And so New Zealand is a very coastal country, but in addition to people often living very close to the coast, often people have holiday homes on some of the more smaller coastal cities. So people would not just see mangroves within the main cities like Auckland and Whangarei, but they would also see mangroves in their coastal place where they would be launching their boat, going fishing, walking around the seafloor. And so mangroves were starting to be seen as a problem species. And so what we had going on in the 1990s and the 2000s is lots of coastal communities got together and they formed estuary care groups. So these groups that were looking at the health of the estuaries, and one of the things that they saw as not healthy was the increase in mangroves. Now, we know now that the mangroves are directly linked to sedimentation. So when we clear forests and remove the things that are binding sediments together, we have lots of sediment erosion, and this sediment erosion into our estuaries is what helps to make places more suitable for mangroves. But back then, people just looked at the symptoms. So they just saw the mangroves as mangroves are unhealthy, we want them gone. And so suddenly we had a whole bunch of consents coming out. And from the science community or university community, most people, if you know anything about global mangroves, you would go, well, why on earth would you want to remove mangroves? Aren't they awesome? Aren't they important? Why would you remove them? And so most of the science community didn't do anything about it and didn't do any research about mangrove removals because they thought it would go away. They didn't think that this public call to remove mangroves was actually a valid call. And so basically you had a whole bunch of things going on in local government, which was approving these consents to remove mangroves, but there was no science to support it because scientists really just couldn't see why there would be any valid reason to remove mangroves. And without that science, that meant there was also no science to show that removing mangroves might cause bad ecological impacts. So we had a number of early consents going on. I have a whole bunch of cities here, and I'm sure everyone will struggle to pronounce a lot of our Maori names, but Mangawai is this first one up in Northland. That was our first big mangrove removal consent. It wasn't very big, about 0.26 hectares. And that removal occurred in 2003. And then there were a series of other very small removals that occurred. And these typically were communities that were clearing pathways for launching boats, clearing areas to build boardwalks or otherwise access the shore, or to allow themselves to be able to see the ocean from the front door of their coastal property. So we had a whole series of these very small scale community removals. And then the big one came in 2010, which was in Tauranga, which is a very large harbor. And this was over 100 hectares. And what happened with this one is they came in and removed over 100 hectares of mangroves using a tractor out on the sand flat, mulching the mangroves. So basically putting the wood through a mulching machine that creates wood chips and shoots them out the back. And they left all of that mulch, all these wood chips on the sand flat. So they basically covered and smothered over 100 hectares of the intertidal habitat in a harbor. And this consent really brought to the forefront the need for scientists to start researching mangrove removals, not just the values of mangroves, but also it spawned a whole bunch of research on the values of mangroves themselves, so that those the ecosystem services that mangroves provide could then be compared against those societal and public desires to remove them. And so we did have a series of another 4 or 5 years of still more and more consents, and I'll show more of this in a moment. But right now in 2020, we're at a very different stage where a lot of the research about mangrove ecosystem services, as well as about mangrove removal impacts, now exists, and that's resulted in a lot of our regional councils having very defined rules about what is allowed for mangrove removal and what is not. And so it's been quite a change since the 1980s, going through a period of everyone in New Zealand thinking mangroves were very important, to a whole period of about 20 or 30 years where people wanted mangroves removed, to where we are now, which I believe is a bit more of a balanced approach. And we've had a large number of research projects, including Suyati's research, that has really contributed to this. So what do we know about mangroves in New Zealand? Well, unfortunately with mangroves, they were these muddier habitats, and most people didn't actually like working in them. They tended to be primarily in the upper North Island, which is a less habited area. So you have less human interactions with mangroves up there. And so we didn't actually have a lot of research about mangroves in the 1980s. But so that meant much of what we know about mangroves, we actually took from the tropical mangrove literature. So in a lot of the early consents of people wanting to remove mangroves, blue carbon wasn't very interesting then. Nobody knew anything about blue carbon, but people knew that mangroves were supposed to be great fish habitats. So this idea that mangroves were fish nurseries and that we had lots and lots of invertebrates and fish and birds. We don't have any native mammals other than bats, but basically these huge coastal ecosystems that supported a whole bunch of extra fauna and flora. And that was what people thought mangroves were in New Zealand. But when we actually look at mangroves in New Zealand, they actually don't really provide a lot of those roles, particularly that role for fish and as nursery habitats. And some of the latest research actually suggests that areas are harbors that have lots of mangroves actually have less fish in them. So it's actually quite interesting to see that the changing idea on whether or not mangroves are valuable for fish in New Zealand has actually shown a complete reversal in suggesting that mangroves are actually very poorly associated, particularly with our commercial fish species, and that they don't serve that mangrove fish nursery role. There's been a little bit of work on the value of mangroves for birds, and much of it relies on the fact that we tend to have built up a lot of our coastal habitats. So we no longer have on the terrestrial side the types of salt marsh or terrestrial forest that used to be adjacent to the coast. And now these mangrove forests are often the only tree habitat around the coastal area. So they then have a much larger value for birds than they would normally. We don't have any obligate or birds or invertebrates or fish. So we don't have any species that can only live in mangroves in New Zealand. And when we look at particularly the benthic invertebrate communities, so the crabs and worms and shells that live there, they tend to be very similar animals that are also found outside in the mudflats and sandflats. So we might have slightly different types of animals, but none of them only live in mangroves. They all live somewhere else as well. We do have lots of things that are researched very well, and I'll show you some of these because they were done by some of my graduate students, but particularly on blue carbon, and on primary production of our mangrove forests that suggests that our New Zealand mangroves are very much on par with tropical mangroves, noting that they tend to be smaller species. One thing to note is our New Zealand mangroves do have a huge range in height. And so in the upper north, in the subtropics of New Zealand, we may find mangrove trees that are up to 6 or 8 meters tall. And around Auckland, we'll still find tall trees that are up to about 4 meters. And as we get to the further south near 38 degrees latitude, in our Bay of Plenty area, often the mangroves are not much more than about 1 meter or 1.5 meters. So we do have a latitudinal range as far as people are aware. On the other hand, we do also find what we call hobbit mangroves. And so if anyone is familiar with the Lord of the Rings movies, the hobbits were the that little shorter group of people that the Lord of the Rings stories are about. And we have hobbit mangroves in New Zealand. So these are these much smaller stature or dwarf mangroves. And so in Suyati's thesis, he looked at both the tall stature or those greater than 1 meter tall up to 6 or 8 meter tall mangroves, but also where do we have short mangroves coming in, and then looking at these changes in how many more short mangroves versus tall mangroves are we getting as mangroves are increasing in their distribution. And I'll show some pictures of that shortly. But in short, much of what we knew about mangroves in New Zealand was based on what people thought the values of tropical mangroves were. And we found that some of those actually work quite well, and some of them don't. So blue carbon, I think, is one of the things that we probably know the most about in New Zealand mangroves, though we're still wanting to learn more. And these are just an example. These are some collaborators here collecting deep sediment cores, so 1 meter deep cores for blue carbon measurements. And this was a project we did with Deakin University in Melbourne, Australia. But most of you will know that our mangrove habitats are very good at carbon sequestration. This is no different in New Zealand. We do have good levels of carbon sequestration, and a lot of this initial research was done by one of our other PhD students about the same time that Suyati was here, Richard Bulmer, and his PhD was on carbon and nutrient cycling and carbon stocks in New Zealand estuaries, particularly in mangrove ecosystems. And he created a whole bunch of this base literature on how much biomass is there for different tree heights. So through a whole bunch of sites, he was able to get measurements of both above and below ground biomass. And he spent far too much time with all these different woody bits and leaf bits and everything else to get these biomass ratios, so that we actually have good equations, not just for biomass versus tree height, but also for carbon and for nitrogen with varying tree heights. And so we've got these for tree height, for trunk circumference, for canopy area, et cetera. And that means when we have aerial photograph estimates, which I think is one of the things that you're learning how to do in this course, that you can then take these photograph estimates and know the tree height and then translate that into the expected blue carbon provided by those habitats. So this was a lot of Rich's research on creating the data to support these allometric equations. And also, like everywhere else where mangroves are found, but something often the public doesn't understand is that a lot of our mangrove biomass is underground. So this was just across a whole bunch of different sites where we actually measured the below ground or root biomass underneath. And here, typically on average, we're somewhere between 1 to 2 times as much biomass as underground. And at sometimes we had 8 times as much biomass underground. So a huge amount of our biomass is underground, which means a large amount of our sediment or of our carbon stocks, or that blue carbon of mangroves, is actually formed and kept in the ground. And that is really why mangroves are so efficient as being a blue carbon habitat, because so much of the biomass of them is underground in these less oxic, or there's less oxygen in these habitats. And that means that you'll have less likelihood of turnover and release of the carbon from the sediments. So that promotes that long-term storage of carbon sequestration in these mangrove habitats. What we've also done, one of a further project we did with Rich, and I should also point out, Rich also shared Lutgard Schwendemann as a supervisor, and that was also Suyati's lead supervisor at the University of Auckland. So she's been part of many of these projects as well. But one of the things that we wanted to look at is how important are mangroves for blue carbon compared to other habitats. So for example, one of the things we often see is that mangroves might be fantastic, but then we don't know how salt marsh or seagrass or even unvegetated sand flats are contributing when it comes to blue carbon. So one of the studies we did right as Rich was finishing up his PhD is we went out and we put together 4 different transects through different habitats. And this is Tairua Harbor, which is on the Coromandel Peninsula, so a bit to the east of Auckland, the main city in New Zealand. And here we were looking primarily initially to see how mangroves compared, but we did get some surprising results from this one as well. So what was nice to see is that yes, mangroves and salt marsh. So salt marsh are the red bars, mangrove are the orange bars, and then our mudflat and sandflat are that lighter green and the blue bars, and the green bars are seagrass. And so generally we did have higher blue carbon storage at all our salt marsh habitats. So usually between 2 to 4 to 5 times higher biome or blue carbon in salt marsh than our sandflat habitats. And our mangroves were usually 2 to 3 times higher than our sandflat habitats. But then we also found some other surprising things, that seagrass wasn't really any better than sandflats or mudflats. So seagrass in New Zealand is typically an intertidal species. They're often a bit sparse and they don't have a huge root biomass. And so that actually isn't terribly surprising when you know the ecology of these types of habitats, that they weren't, at least in this case, providing any more blue carbon than adjacent sandflats. But then one of the other things we did with this study is we looked at the total area within the estuary that was in each of these habitats. And when you look at over 80% of the total habitats is sandflat, so even though sandflat might be a smaller amount of blue carbon contribution than salt marsh and mangrove, because it's such a large portion of the total estuary, it is actually the largest contributor to blue carbon in most of our New Zealand estuaries. So this is something we're looking at now. We just got a new grant that we found out about a month ago, where we'll be looking at blue carbon in more depth across different habitats and trying to see if this pattern holds in all of the estuaries in New Zealand. So one of the other things that we know quite a bit about in New Zealand is the trophic role of mangroves. And so around the world, we see mangroves as having very high primary productivity, and that leaf growth basically fuels food chains. We have the leaf litter falling to the ground, and then through that decomposition and things, crabs and other animals eating it, that it quickly becomes part of the food web and fuels these fantastic tropical ecosystems. Well, this was the assumption of what was going on with mangroves in New Zealand. And this is the work of another one of my former PhD students who also did a master's with me. And she was actually at the University of Waikato here in Hamilton. And what she was looking at was leaf litter fall and decomposition of mangrove leaf litter and how this affected changes in the benthic invertebrate community. And so I think the first thing is, when we looked at it, we did measurements of mangrove leaf litter fall. And not surprisingly, we have a big seasonal contribution of when the leaf litter falls, which tends to be in our summer. So that's November, December, January is our New Zealand summer. But that and then this pattern is consistent. We've since looked at about 8 other sites and have done a whole latitudinal study, and we don't see any latitudinal differences in mangrove leaf litter fall either. So it's pretty consistent across all of our species in the amount, and the amount in terms of litter fall is comparable to the same species and a number of other species on the low end of the total mangrove primary productivity, but within the range of primary productivity of mangroves around the world. We also looked at how quickly does the mangrove leaf litter decompose? And this is one thing where mangroves in New Zealand are very different to the rest of the world. So looking at both the decomposition of leaf litter, but also of wood and pneumatophore. And these things are quite relevant also to blue carbon, because if we have very low decomposition rates, then we'd expect that long-lasting blue carbon or carbon storage in the sediments as well. And in general, in a lot of the tropics, the leaf litter decomposition is often within about a week. We looked at our leaf litter, and this study was just at one site, but we've again repeated this experiment at 8 different estuaries from the far north down to the Bay of Plenty, so through the New Zealand mangrove distribution. And we have a pretty consistent decay time of 2 to 3 months to the decline in half of the biomass. So our mangrove leaves decay very, very slowly in New Zealand, and that means they're very slowly incorporated into the food web. We also don't have any major grazers on mangrove leaves either. So we're not seeing huge grazing of any gastropod species, of any snails or crabs. And so we basically have the leaf litter deposited, and it takes a very, very long time to be incorporated into the food web. Now, that doesn't necessarily mean that we don't have any changes in community. We did some experiments looking at the benthic community, and there were slight changes, but they're very minor changes in benthic community based on this addition of nutrients from mangroves. So I've got one more slide up here on some of our mangrove ecosystem services. Now, I started the talk telling you about how people want to remove mangroves in New Zealand. Well, this is one place where people don't want to remove mangroves. You can probably see in this picture that there are a whole bunch of very strange lines of mangrove trees, and these are all planted mangrove trees in front of one particular landowner's property. And so these are actually kiwi fruit vines here. And this landowner was seeing lots of erosion from storms. And so he decided he was going to plant a bunch of mangroves and see if that made a difference to the erosion of the land near his property. And hopefully this picture, it may not show up very well, but these are actually the waves coming in, and you can see the wave height. Hopefully you can see this. I know it's quite dark on my screen, but the wave height, you could see very significant reductions in wave height of the waves coming in, and thus a reduced impact on coastal erosion where he had planted the mangroves. So in some places, we do have landowners recognizing the value of mangroves and using them for the ecosystems they provide, like this coastal protection value. And up in Auckland, we're starting to see a lot more recognition of the value of mangroves in preventing coastal erosion, so that there's a lot less interest in removing all mangroves, and instead we're having a lot of requirements to retain buffers along the land so that we don't have this significant coastal erosion. But as I said, mangroves are expanding in New Zealand, and they expanded primarily from the 70s through the early 2000s was kind of the main pulse. I've got a picture here. This is Whangamata Harbor. So this is, if you see the star on the little inset map, this is on the Coromandel Peninsula on the North Island. And this is really a beach or a holiday town. So most of these towns here don't have permanent residents, and they're all people going out there for summer holidays. And what you can see is in white, those would have been the 1944 distribution. So about the first time aerial photographs were available during World War II. And then we have that further expansion in blue in 1978 from an aerial photograph image. And then in 2002, this is more or less the current distribution is shown here, including the white, the blue, and the orange is the more or less the current distribution. What's important to see here is that initially a lot of the mangroves would have been in the upper tidal creeks and not where any of the community could see them. And then as you could see, as this change in mangrove area in the 1980s and 90s, it's really come in around where people had their homes, and it became actually quite apparent to people that the mangroves were expanding, whereas the mangroves up here in the 1940s weren't really where people could see them. And that's quite typical of how mangroves are expanding in New Zealand, is it's this new conflict with human amenity values, or where society originally had views of the ocean or views of the tidal creek, and now there are mangroves there that have changed how they feel about the ocean because it's not the same as it was 20 or 30 or 50 years before. We do see huge differences in the societal preferences, whether someone is older or younger, in that younger people would have grown up with mangroves there and tend not to have the negative values toward mangroves, where a lot of our older communities, particularly our retired people, knew what the place looked like beforehand and aren't quite as happy to have mangroves there. Anyway, I've got just an example of some of the increasing spread. This is from one of Suyati's papers, and this was across the Auckland region on average using aerial remote sensing. So the satellite images that were available from the Auckland region, and you can see that we had that reasonably consistent increase in, again, 3.4% per year on average in the Auckland region. However, it's quite different when you look across different types of mangroves. Here, just looking by stature, so that the white dots here are our tall mangroves, so things that are taller than a meter that people would recognize as being a tree and having a good value in terms of the ecosystem services that they're providing. And the increase in these tall mangroves is really only about 1% per year. In contrast, these dwarf mangroves, so these much shorter mangroves on average have been increasing quite rapidly, up to 20% per year. So it's those small mangroves that often people don't see a huge value in that are having much more of an increase than the tall mangroves that might have more ecosystem services that they're providing. So why are mangroves increasing? Well, I think initially people thought mangroves were the ones that were causing increase. So is it the mangroves or is it the sediments? So do mangroves come in and colonize everywhere, and then they create more mud by creating conditions where mud is much more likely to settle? Or do mangroves just only come once the mud is there? Well, it's actually a bit of both. And so initially, I think I've got a slide coming up, hopefully, of some work by a colleague at the University of Waikato, but we've also had a lot of work done by a colleague at NIWA, Andrew Swales, and he did his PhD at the University of Queensland. And what he looked at was these very long, deep cores and using radiocarbon isotopes to look at sedimentation rates at different ages based on where mangrove or how old a mangrove forest was. And we have one of our mangrove forests in the Firth of Thames. I think it's about, 2 or 3 kilometers wide by maybe 40 or 60 kilometers. So it's a massive mangrove forest. And my understanding, it's the largest continuous mangrove forest in the Southern Hemisphere, but I should check on that. But a beautiful, huge mangrove forest. And when James Cook came to, one of the kind of early European explorers, when he came to New Zealand hundreds of years ago, this mangrove forest did not exist. And so it's actually a new mangrove forest that we can actually track over time, the age of the mangrove forest and when the big recruitment events occurred. And it tends to, this particular mangrove forest, roughly every 10 years, it would have a big recruitment event and extend out another 100 or 200 meters. So looking at this particular mangrove forest and these deep, long sediment cores, what Andrew found was that the sediment rates after the mangrove forest was established weren't any greater. However, some more recent research has actually looked at that fringe area, or where the pneumatophores are in mangroves, and has shown that the pneumatophore zones themselves do actually enhance sediment deposition, so that we are seeing more muddy sediments being deposited here. And it's these pneumatophore zones that tend to be the areas of expansion of mangroves. What do I have next? So why are mangroves expanding in New Zealand? Well, the main conclusion that it's really changes in sediment runoff and sediment deposition in estuaries. So when our sediments come down, we have very steep catchments, and we have a lot of changes in land use, both clearing of native forests for urban development, but also for pastoral or agricultural uses. And these things tend to result in sediment erosion from land. The sediment washes down the streams and it's deposited in our coasts and estuaries. This means while we're having the negative impacts of sediment by smothering the sediments and creating more turbid water, it also is a bit of land building. So we have sediments coming into the estuaries and gradually increasing the amount of intertidal habitat through the infilling of our estuaries. And that's our main locations where mangroves are expanding. And I'll show you some pictures from Suyati's thesis that show this quite nicely. We also have some other places where we have changing hydrodynamics. So for example, when we put in marinas or motorways, so this picture in the middle, you can see that we have this, this is our main motorway number 1, and you can see that it's changed the flow, and suddenly this entire small embayment is completely full with mangroves. And we've seen that in many areas where we change the flow dynamics and these areas become cut off and fill in with mangroves. On the other hand, one of the things we haven't really seen in New Zealand is the type of mangrove expansion we're seeing in a lot of other temperate places. So for example, in Florida, where a lot of the mangrove changes are moving shoreward, so that things are moving inshore toward land with sea level rise, or colonizing into salt marsh habitats and taking over salt marsh habitats. This is something to date that we haven't really seen much in New Zealand, and possibly it's because no one's looking. So that's another thing that we're hoping to do quite a bit more work on in the near future. And then we also haven't seen any changes in range. So there's a chance that New Zealand mangroves could move to the south, so further south than 38 degrees latitude. We haven't seen this yet, and it's possible that the frost limits will be changing in the near future as areas have less and less frost damage. right now in the Bay of Plenty is where we start to get the coastal frosts that would change the survival conditions for mangroves. However, it's unlikely that we will have significant range shifts because we really don't have a lot of estuaries then for about 100 kilometers at least to the south of where the current mangrove distribution is. So quite likely these range shifts aren't going to happen in New Zealand because the available estuary habitat just isn't there until we get quite a bit further down on the North Island of New Zealand. So it's probably not possible just due to that distance between our estuaries and harbors as you move south. So I wanted to show off a whole bunch of Suyati's works, and you'll know him as one of the instructors in this course or organizers. And what Suyati did in his thesis at the University of Auckland was using our satellite remote sensing photographs then to both take the satellite photos as a measure of where the mangrove distributions are, as well as look at that across time and compare that to a whole bunch of climatic drivers that we had. So both our changes in temperature and other factors that we could find in these large databases, but also looking at changes in land use. So what type of land cover was there? When did land clearing happen? And how were those factors then influencing when mangrove expansion occurred? And what do we have? So we had some really nice figures that show the different types of ways that mangroves were expanding in New Zealand. And these vary from gap filling. So in places we might have just a few trees and they are gradually infilled. This is one of the very common ways of mangroves expanding, but also seaward expansion. So most of our mangrove expansion in New Zealand, as I mentioned, is seaward. So we have more sand being deposited or mud being deposited in estuaries. It's increasing the intertidal area through extra kind of land building, but as you're going seaward, which again is quite unusual in the world. And so we see mangroves then having new habitat that they can colonize as the infilling of estuaries creates more of this shallower habitat that's within the tidal zone that they're able to live in. We do see occasional landward expansion, but it's not as common in New Zealand. We also have upriver expansion. So this would be where a lot of our tidal creeks are gradually colonized by mangroves, and also some other transition zone type things here in particular, where we see changes from these tall mangrove habitats to being replaced by these dwarf mangrove or those hobbit mangroves. And then these are just some examples of a few of these from particular areas where you can see that kind of 1940 would be on the left, in the 1970s in the middle, and then more current distributions here on the right. So you can see that change if you color code it. Good, I do have it on here. So our color coding would have mangroves that are the tall mangroves are in the darker red, and the short hobbit dwarf mangroves are in that lighter, green, the two different green colors. And you can see this change particularly here where we once had a lot of these larger mangroves and they're gradually getting taken over by these smaller mangroves. And we're just seeing the large mangroves often still remaining on the edge of our main channels where we have a lot of water flow, but that we are seeing this change both in increasing in mangrove distributions, but also a change where we have more of these shorter mangroves and less of the taller mangroves. And again, this is a reasonably consistent pattern through much of New Zealand, but certainly the Auckland region. And then just a couple close-ups just showing you some of these types of patterns. And what's important is that these do have huge implications for ecosystem services. So if we're changing from, for example, these gap filling where we have the odd mangrove that probably isn't providing a huge role in either carbon storage or coastal protection or other ecosystem services, to one that's a very well connected habitat, that's one case where this is very good for ecosystem services. On the other hand, if we're seeing the change being from very tall 3 or 4 meter mangroves being replaced by 60 centimeter mangroves, then we may actually be seeing a very large decrease in the ecosystem services that are provided by mangroves. What else do I have in here? So then just this next slide again was just trying to showcase that it's really this seaward transition. So mangroves moving seaward are much of what we're seeing in New Zealand. That's quite different from what we see in the rest of the world. And then I think just some of the statistics on comparing to those climate drivers, as well as that change in land cover, and some of the statistics that Suyati did showed that over 50% of the variability in mangrove expansion was directly related to that basically clearing of catchments that are then associated with increased sediment erosion. So it's really that sediment erosion in New Zealand that's helping our mangroves to find new habitat to expand into. So great for mangroves, but again, bringing it back to the wonderful world of politics in New Zealand, not always good if we have local communities not wanting mangroves to be there. And so early on, what we had was just these tiny small removals, and often they were quite successful. So we would have a community group going out and removing 10 or 20 trees, so not a very large amount. And these are a couple of the places where we went back to the early removals 10 years later. And if you look at them, you can see we have healthy sediment profiles. So we see this nice tan sediment. We have bivalves living there. We have crabs. We have gastropods. So a healthy benthic community, even though quite often there's still strong evidence that it was once a mangrove forest because the stumps haven't decomposed. Other places where mangroves were removed, this picture here was one of the first government consented mangrove removals in 2003. And you can see it's now a mangrove forest again because the mangroves colonized pretty much immediately thereafter the mangrove removals occurred. And most of our mangrove removal activities have never had any monitoring or any maintenance. So often a community group would get a lot of excitement. They would get people together, the mangroves would be removed, and then nobody would ever look again until someone went out again 10 years later and went, oh, the mangroves are back. And so then just this real challenge of knowing whether or not areas that mangroves removed were successful. Those people that were anti-mangroves would often remove or promote the evidence where things like this happened, where everything looked very good. But if things didn't look good, and if they had any environmental impacts, these often weren't ever publicized. So there was no evidence to suggest that mangrove removals could be a bad thing. And so I think just a couple years before Suyati came to New Zealand, we started doing some direct monitoring of this big mechanical mulching. And I've got a picture of the beast doing the mulching here in Tauranga Harbor. And you can basically see this tractor was going back and forth, removing mangroves and then spitting all the debris out the back and basically grinding it into the sediment. And I think any ecologist, if they'd known what was going to happen, just would cringe. And I don't think any of the pictures that I have coming up are nice and more shocking that this type of thing would happen in New Zealand. But what people were worried about with removing mangroves was not the benthic habitat or whether the estuary would have enough oxygen or whether there would be smothering. No one really cared about that. They actually were concerned about shorebirds primarily. So was this going to have an impact on birds? Would the sediment move from these sites and bury the shellfish beds that were neighboring around? And so really what people were looking at monitoring actually had nothing to do with the impacts that were actually seen. So there was a big mismatch between the types of monitoring that the government consents required and their ability to actually detect impacts because they weren't monitoring the right thing. So we as researchers came in and did a whole bunch of research on impacts that was then used to basically tell them it was a bad idea and then was used by all the additional consents for mangrove removal in New Zealand. And so here's some of, these are my horrible pictures. And a lot of these are actually a number of years after the original mangrove removal. This was again in Tauranga Harbor in our Bay of Plenty, and it was about 100 hectares that were removed. But we had significant vehicle tracking. A lot of the places you can still see the vehicle tracks, and this is, most of the removals were in 2010 and 2011. We had a lot of major algal growth. One of them, we found a new record of an invasive algal species. So that was kind of exciting, but not really what you want in your harbor, particularly when it was such a huge area. There were a lot of complaints by the local communities who'd advocated for mangrove removals because then they had a persistent hydrogen sulfide odor over the course of 4 or 5 years. So it significantly affected people's enjoyment of the estuary, and it affected recovery. And one thing it didn't affect was the ability of the mangroves to recolonize. So you can probably see here, we had a whole bunch of mangrove seedlings still popping up every year. But of the challenge with what was done for the official monitoring suggested that there were no impacts whatsoever. And so if you look at my pictures here, you might find that hard to believe, but that was the story that the regional council presented, that there were no impacts and that it was a very good idea. And so then we had a couple other very large consents where a similar thing happened, where they did this mechanical mulching. We had the same exact results. We had very degraded habitats, and these continued on for a number of years. And these were both, this was Auckland Airport, which, to be honest, I think most of us supported this one. We were having black swans roosting directly next to the runway at Auckland Airport. So for health and safety purposes, they removed the mangroves to and then get the black swans to roost somewhere else so that they weren't having a hazard to planes and having planes hitting birds and then crashing. So that one actually did have a very different reason for going forward. On the other hand, it provided the same exact type of impacts. Oops, I scooted forward a bit too quickly. But the general consensus was that most of the scientists knew mechanical mulching was bad, but the management agency that was in charge of the original mechanical mulching at a different site wasn't monitoring the right things and showcased that something that didn't have an impact was. So we were able to fortunately get the information across that this particular method was a really bad idea. And then we managed to get additional funding then to look at 40 different places. I think this was in 2013. So we went from the Bay of Plenty. So way down here is where Tauranga Harbor is. I think I've talked about Whangamata and Tairua are a couple of the places we often go to. Auckland is right here. This massive mangrove forest in the Firth of Thames is right down in here. And then we went all the way up to Whangarei Harbor. We didn't go quite as far to the far north, just due to logistics. And we hit 40 different places where mangroves had been removed to look at how did different methods that people use. So if people used chainsaws or if they used tractors or what types of methods they did that actually have an increased likelihood of having a healthy environment after mangroves were removed? Did the mud disperse? Were places recolonized? And we had locations that had mangrove removal activities anywhere from, I think it was about 3 months to as long previous as 16 years. So looking across all 40 of those locations, in general, we had, I believe, 3 sites in total that had recovered to a healthy state, but in general, very few of them ever transitioned to what was there 50 years before, which might have been this more pristine intertidal sand flat. So our communities that are wanting mangrove removal figure if they remove mangroves, they'll get this pretty beach in return. And what we showed was this actually wasn't happening. And there were very few places where we were actually seeing mangrove removal working. That said, that didn't stop the communities from wanting to remove mangroves. And again, here, just some of the examples of some of the methods that people would use. Here's again that tractor. We also saw people collecting piles and doing burn piles. So of the very strange experiments that people have done, we know exactly how much carbon is released by a burn pile of a certain height and density for mangroves, because that was required for the aerosol emissions for one of the consents. We also had some places that used a helicopter removal method, which again is fascinating to see the lengths that people in New Zealand would go to remove mangroves. But here we basically had a consent where someone hired a helicopter to basically bundle up all of the above ground biomass. So all of the branches and stumps were bound together and then brought out by helicopters. So quite interesting what people were doing, and showing, kind of the big difference was if the biomass was taken off-site, you tended to have a much higher likelihood of a recovery to a healthy environment. And what else was, but also how much flow you had and the original density of trees were also places that were typically associated with very strong recovery. And this is just showing 3 of the places that actually did recover quite well. I'm not sure if you can see too well here, but all of these pictures do have stumps in them. So they were, I think the stumps are probably the most apparent in this picture on the bottom right, where you can see a lot of them on the bottom. So this was a fairly large mangrove forest in all of these cases. And so we had very few of our sites actually showed any recovery after mangrove removal, even to a healthy state. And rather, most of our sites look something like this, like some of the pictures that I showed you just a few slides ago. So where we had that consistent deposition of this material. And I'm trying to think, I'm not sure if I've got a picture of one of these sites that we've since visited nearly 10 years later, and still a very degraded condition, and in general supporting that mangrove removals generally did not have very high success rates. This is just another one where we're showing, this is actually on the side of the motorway in Auckland, and this whole area was a mangrove forest that was removed. And this is actually an algal growth. So that's a ulva species, I think, on here. And oops, and my line has moved, but that line should be right there on the side, which just shows the difference between, this is the little grass edge of the motorway, and everything over here is actually showing this nutrient addition and the rotting biomass from the below ground mangroves that are still there. And this still looks reasonably horrible today. I drive by it every time I get to drive up to Auckland, and it still is in a reasonably degraded state. And that one, again, that was in 2012, so it's nearly 10 years ago. Some of the other things we learned about mangroves. So here's a typical, what we'd call an illegal mangrove removal, where often communities would be frustrated about mangroves existing, and they'd actually just chop the branches off and just leave all the foliage there. This happens quite frequently in New Zealand. And one of the things we found is that mangrove stumps actually do resprout as long as they don't have any entry points within the waterline. It's probably worth mentioning that I didn't earlier that our tides are reasonably small in New Zealand, so most of these sites would have a 1.5 to 2.5 meter tidal range. And so a lot of our mangrove sites really don't get water much deeper than about 30 or 40 centimeters at high tide. So we're not seeing like the great tides like you see in Darwin, Australia. I think it's an 8-meter tide, and you have these huge mangrove root structures that animals can live and swim among. Our mangroves tend to have a very small amount of water coming in when the tide comes in. And so because of that, that meant that this particular tree was actually able to resprout after most of the branches had been chopped down, because it wasn't actually getting the salt water into the main part of the tree. So a little bit of ecology that we learned, but again, and I think these are all 5 years on to a bunch of these sites, that they're all still in reasonably horrible shape. And I think the one we go back to regularly is Waikaria, where we've done a whole bunch of field experiments, and it looks a little bit better than it did, but still certainly not wonderful. But they don't have quite the same problems with the algal growth and the same issues. That's finally disappeared after 5 or 6 years. So does mangrove removal result in the muddy sediments going away? This plot is probably quite difficult to look at, but basically each chunk of 3 or 4 blocks would be our mangrove sites. So we had mangroves, the center of removal, the edge of removal, and the neighboring sand flat. And for the most part, you can see everywhere that we have mangroves and mangrove removals that they tend to have very similar sediment muddiness, that we're not seeing a lot of disposal or dispersal of the muddy sediments. And so once the mangroves are removed, the area tends to stay as muddy as it was. And this makes sense to us because most of the time the sediments that colonized there or were deposited there tended to be very muddy sediments before the mangroves came in. We're also seeing, not surprising based on some of the studies we did on decomposition of leaf litter and of root and woody material, that the decomposition of the below ground material is very, very slow. So we often saw this persistent material certainly lasting at least 10 years, if not longer. So very high amounts of below ground material. And if those are still there, it makes sense that they're still binding those muddy sediments and preventing their dispersal. So not terribly surprising that after mangrove removal, it takes a very long time for any change to occur. So is it working? That's the question we always get asked. Does mangrove removals work? That's for people who want it to work. Basically, our sheltered and muddy sites typically didn't show any change at all. We tended to still have very high mud content, high below ground sediment or mangrove biomass, and really not any positive changes in the habitat, that they not only were still a mudflat habitat, they were still quite a degraded mudflat habitat, and they certainly weren't showing any sign of becoming any sandier. We had a few exposed sites that tended to have very high exposure to either wind waves or tidal currents. These were the ones that were most likely to show any change towards sand flat, but still quite slow changes. So as all of this was going on, we had one of the councils, so this is one of our regional management agencies and the one that is in charge of the kind of the next region south of Auckland, our Waikato region. They were looking at doing some adaptive management and a harbor-wide coastal plan in Whangamata Harbor. So, and they asked if we could help them design a mangrove removal plan to see if they could actually make it work and minimize the impacts. And a lot of this was, could they remove mangroves, because they were realizing that if they didn't let the community group legally remove mangroves, that they were going in and illegally removing them and creating far more damage. So it was basically in response to illegal removals that the council opted to go for a mangrove removal. And they came up with this idea that, well, is it the mangrove or the tracking of the tractors that's making the most damage? So could we just have a tractor come in and have the arm of the tractor stretch out 10 meters, remove mangroves, take all the biomass off site, or in this case, they initially left it in these burn piles, and see if they could actually come up with a way to remove mangroves that actually had a positive outcome. And if you're looking at this picture on the bottom right, that's 5 years after the mangrove removal, and you can't tell the difference between that and a sand flat. So we basically showed that you could work. It's still a slow process. You do have a very slow reaction. This was in total, I think this consent was about 20 hectares, and they, I think, only actually went through on about 10 hectares, and then they ran out of funding. But basically, there are ways you can minimize those adverse impacts, primarily through that removal of the mechanical disturbance. and then just the awareness that it does still take a long time, so that a lot of the consent documentation that we came up with was actually looking at trends in different metrics. So trends in removal or changes in muddiness, but also making sure that they avoided. You'll notice that you don't see the anoxia outside of these main channels. So most of the places where the tractor went, we have that black sediment where things were anoxic, but beyond that, we did have healthy sediments. We didn't have algal blooms here. We didn't have the bacterial mats. We didn't have the hydrogen sulfide area smell. So a lot of these things that they were actually able to avoid. Still doesn't answer the question of whether you should remove mangroves, but as part of the experimental work we did with them, they showed that you can remove mangroves and have a change to a sandflat habitat, as well as a minimizing of the environmental impacts. And so here, just a couple more pictures of these sites and what they look like about 5 years later. So quite different, kind of 2 years versus 5 years versus, and all of these would have been kind of 2 meter to 2.5 meter trees, very high density mangrove habitats. So it did work. Again, doesn't matter whether, or doesn't answer the question whether we should remove mangroves. And at the same time, we also looked at what else do we need to worry about if we're removing mangroves? So early in the talk, I mentioned that quite often we'll have a mangrove removal activity occur in New Zealand, but we won't have, oops, sorry, greeted by a dog. We won't have any of our, any long-term monitoring. So this was this first mangrove removal that within 10 years had been fully recolonized. This site here is another one of our study sites, and that one was recolonized within a couple years of the mangrove removal here. So in many cases, our reasons for mangrove removal are actually quite legitimate. The one here on the right, this is actually a cultural heritage location. And in when Maori came to New Zealand, about 1,000 years ago, one of the original passageways was from the east to the west coast, where right around Auckland, we have a very quite thin spit of land that's just about 5 kilometers across. And so in the waka, or the ancestral canoe that came over to the east coast of New Zealand, they actually carried this canoe across Auckland and put it in Manukau Harbor, and right at this location. So this location in the picture on the right, it's actually cleared because every 2 years they have a celebration of that ancestral canoe going across the land border from the west, or the east to the west coast of New Zealand. So this is a cultural heritage site where they remove mangroves, but it means that it's somewhere where it's part of the culture of New Zealand to maintain that passageway so that the canoe can actually go out through that environment. A lot of our other mangrove consents are for motorways or for accessing flood protection zones, for power lines. So we do have a lot of reasons where we actually need mangrove removal to occur. And the vast majority of our mangrove removal consents in New Zealand are actually for infrastructure or other types of requirements, and they aren't for societal perceptions or recreational values. And so one of the things then at the same time that's also important is to recognize how much funding was required for councils to maintain a line of mangroves, so that we're not having this constant need of, if they did have adult removals of mangroves, how much would it cost to actually maintain that line of removals from seedling removal. And so we did a seedling removal experiment with a local community group, and it was building on some research that I mentioned my colleague Andrew Swales. He did some early research close to 20 years ago now, looking at survival rates of seedlings in the Firth of Thames. The Firth of Thames, that's that big, huge mangrove expanse in a firth, so it's a very unusual environment that has very, very high wind fetch. And what he showed was that less than 10% of the seedlings survived 2 months, less than 1 survived through that kind of summer season. So typically we would have years, for example, I think it's our El Nino years or La Nina years, I can't remember which one in New Zealand, where we have these periods where we would actually have 2 or 3 or 4 weeks with very low winds. And when we had a year like that, roughly every 10 years or so, we would have these huge recruitment events in the Firth of Thames. But the vast majority of the time, we had very little survival of mangroves. So you had very little expansion, except these kind of one-off years about once a decade in the Firth of Thames. So that natural survival of seedlings was seen to be very low. And was that really the case everywhere else? Because most places weren't like the Firth of Thames. Most of the places we have mangroves growing are in upper tidal creeks, in sheltered bays, in sheltered harbors. So if we're not having these huge winds and waves that are likely to dislodge the seedlings, what is their likely survival rate? So we did an experiment with a local community group. They were excited because they wanted to remove mangroves, and this actually let them remove mangroves. At the same time, it let us as scientists work with them and help them to understand how mangroves worked. And I think it ended up with them no longer removing mangroves. So it was quite an interesting experiment, but we set up an area where they looked at seedling recruitment. We went out once a month over the course of 2 years, so about 25 times that we went out with much the same people, and had over that course of 2 years probably had 200 or 300 different individuals come and help us. And we counted and measured the seedling recruitment over time. We had 2 different sites, and we looked at seedling growth over time, or seedling survival over time. So I would have showed you that Firth of Thames is that blue line of survival of that really highly windswept environment. We had a sheltered and exposed, kind of a more typical New Zealand site of our 2 sites. And we saw instead 40 to 60% seedling survival in an average summer. So that suggests we have something very different going on, and a high likelihood of this rapid colonization, which is what the community groups saw and what the councils weren't believing them that was occurring. And I think I've just got some pictures of us working with these community groups. You know, some of these are right at the end. You can see we've got these meter and a bit tall mangroves by the end of the 2 years, whereas early on we just had very tiny seedlings, and we had kind of great action or participation by the different community groups, all of them learning how to walk in the mud. I'm not sure how familiar everyone in the audience is at walking in the muddy mangroves, but if you haven't done it before, it can be quite uncomfortable, of going squelch and getting stuck. So, but it was a great learning experience, and we had quite a large number of people come through to learn about mangroves. And, you know, often many of them changed their perceptions on what they thought the value of mangroves was. We were able to see exactly when our seeds come in, as well as when the seeds grow. So we know most of our seed come out in our summer period, that December, January, February. And then we also were able to see exactly how fast they grew. And the general idea for mangroves is people assumed it probably took 5 to 10 years for a tree to grow. If you look at this picture on the bottom, these pictures are all more or less the same exact place. I unfortunately have slightly different angles for all of them, but if you can see this fence right in the back here, this fence is in all 3 of the pictures. So you're basically just a picture of the same exact place. This is on the left is when we started the experiment. You can see a few little seedlings here and there. Then 3 or 4 months later, you were starting to see a few more seedlings after that big summer recruitment period. And then near the end of the experiment, so this is about 1.5 years in, we actually had, I think our tallest mangrove was 1.4 meters. So they grow a lot faster than people thought they did, particularly in this small harbor, which does have a history of high nutrient loads. And so we were basically working with this community group to learn more about that natural history of New Zealand mangroves, which was something that no one had ever done before. So when do seedlings come in? How quickly do they grow? And what is the natural mortality rate? And all of these things then provide additional information as our councils are trying to manage mangrove expansion, to recognize that we can actually and do often see very small embayments, basically going from 0 to full mangrove cover just in 3 or 4 years in the Auckland region. But it also helps for councils that do want to manage mangrove spread, that we did see about 50% mortality regardless, and then going out, if they wanted to do the actual removal of seedlings, that they could do that in April or May and have less effort than if they removed them in February during the summer season. So what do we know about mangrove removals? Again, sometimes it's still hard to get your brain around the fact that New Zealand has rules for mangrove removals, but mechanical is bad. Manual can still be bad. We definitely don't want on-site disposal of trees or branches or leaves. And really the places that are still doing mangrove removal, where they're doing it successfully, they're keeping to very small, thin strips at the seaward edge on the order of 10 to 20 meters of clearance a year. And that has shown to be very successful. But really the big challenges are that we're still not seeing the improvement in catchment management in New Zealand. New Zealand has a horrible record for what we allow in terms of clearing of land, both in our native forests historically, but also in our timber forests, in what we're clearing for urban expansion. There's a lot of push in New Zealand to create more housing, and so big suburban spread outside of Auckland that results in a lot of changes in land use and a lot of sediment erosion. And then at the same time, the seedlings are still there, the seeds are floating around, and if any area is cleared, that you still need to have that long-term seedling maintenance if you're going to spend the effort clearing a site of adult mangroves. To help out, we've made cute little guidelines so that these have actually gone out to all the councils. And then these were also something that was given to, I think I mentioned early on that we did have a submission to Parliament. So a bill was submitted for mangrove removal. And so we presented to the Parliamentary Select Committee and provided them with a box of these mangrove guidelines. And then the bill was actually tabled, whatever the word for, they didn't actually go forward with that bill to remove mangroves based on the evidence that we presented. So that was quite good. The guidelines had, again, a huge number of people contributing, and particularly I've shown the work of Sue Yachty and Richard and Rebecca, my 3 PhD students, and heaps of other people and councils, and lots of funding that went into all of this that we did. But generally for mangrove removal in New Zealand, seedling removal was really all that we were recommending. It can be done to maintain an area clear of mangroves. It's relatively inexpensive and it has minimal environmental impact, whereas manual removal works in some places, but not in most, and mechanical removal really is quite damaging to the environment and should be avoided at all cases. Now, that may seem really obvious now, but obviously we did have about 20 years of having to do research to show that these things did or didn't work before our government officials were willing to then work based on the evidence that we provided and make decision-making to avoid environmental impacts. So that was just a picture showing some of the sizes and shapes that we were recommending, but that really to have any change in habitats, that kind of that outer band of mangrove removal has been what has been successful. Typically what we'll see is something like this. So the green is a mangrove forest and the brown would be the clearing that someone's made. Blue is ocean and gray here is land. And so this would be quite typical where people will clear an illegal pathway through to the shore so that they can launch their boat. So that's probably the most common clearing. And then we also have a lot of illegal clearings where people clear directly from land. These still go on in New Zealand, but they're becoming less and less. and then our 4 mangrove regions, so the 4 northern kind of council regions of New Zealand are all starting to be more consistent with the types of rules and regulations they have about mangrove removals. So where are we now? So I think we've had huge changes over the last 50 years, where initially people appreciated mangroves because of all of that they were reading in National Geographic and watching on the Cousteau channel and things like that. So people thought mangroves were amazing in the 1980s. And then this really changed to a number of decades where people saw the risk proposed by mangrove presence. And so there was a big push to remove them. And I think now with the changing information and understanding on the benefits of blue carbon, you'll know that the COP26 is going on right now in Glasgow, and in New Zealand's commitments to climate mitigation, we're hoping that our blue carbon ecosystems will become quite a large part of that in New Zealand, but that people are just becoming a bit more aware of the impacts of removing mangroves and looking more at the benefits of mangroves and the ecosystem services that they provide, and less on what's going on in terms of mangroves preventing view. So the latest consent I got asked to review was actually quite exciting, because they needed a consent to remove a 1 meter band of mangroves through a forest so they could build a mangrove forest recreational boardwalk. So it was actually creating recreational access within a mangrove forest to enjoy the mangroves. So there's been a big shift in how people appreciate mangroves in New Zealand. And I think really that's wonderful to see these changes happen. And I think a lot of this has happened really since Suyati has been there, that we're really seeing that huge change. But also there's the recognition that we do need rules about removing mangroves, because a lot of our mangrove removal is for infrastructure reasons. So we do need to have the ability to remove mangroves because they do grow alongside our motorways, electrical lines, and other things. So we do need to be able to actually remove mangroves legally for a variety of reasons that are actually very good for society, but potentially not so much the other reasons that are around viewscape and being able to see the ocean and things like that. So just big changes. And of course, we've written a whole bunch of papers and things about this. And I don't know if the team in charge of the course, I've put a whole bunch of references in here if anybody wants to read them, and feel free to contact me if you can't access any of them, and I can send you a PDF. But basically, what do we need to do for mangrove management in New Zealand? We really need to manage our catchments. We need to reduce that sediment load into our estuaries and harbors. Not only will that reduce the spread of mangroves, but it makes for much healthier estuaries and harbors in general. So anyway, that's me. Again, heaps of people involved in all of the research that I've presented, and particularly thank you to Suyati for inviting me to be a part of your course. So does anyone have any questions?
Yeah, thank you very much. Very interesting presentations for this give us big picture on the temperate mangrove, especially in New Zealand, and this also bring my mind back to New Zealand. So I miss New Zealand and the people as well. Okay, we still have around 45 minutes for questions. Please, you directly or you raise your hand is here, or directly ask your question to Karen. Okay. Diana, number 34.
Thank you. It was a very interesting stories and studies, but I have one simple question. I'm wondering about the short versus the tall mangrove. Is it based on the mangrove species traits, or you classified it in different ways? So I'm curious about that. Thank you.
Are you meaning the difference between tall and short mangroves?
Yes.
Yeah, so completely based on height on the other, and Suyati probably actually knows this better than me. I think it was just a 1 meter height difference. I'll see if he nods. Yeah. And that was, we did have the LiDAR along with the remote sensing. And I know what's available now is quite fancy to look at tree heights. But then in addition, we do see very big differences in density of trees. So I'm trying to think of what pictures, I don't know if I can share the screen again, that'll show you, hold on, some of the very different, did I show the right one? No, I showed the wrong one. Hold on. Well, let me unshare screen. I shared the wrong side. Oops, share. This is the challenge when you've got two different screens and I've lost my mouse. There we go. So I think in this picture, we've got some of a lot of trees that'll be showing up here, but we've got this picture of Rebecca, or no, that's my daughter. So those are going to be kind of 4-meter tall mangroves in this forest that she's in. Whereas the one I'm standing in, obviously I'm about 1.7 meters. So that one's probably about 1.2, 1.4 would be a very, very typical mangrove forest in the Southern Bay of Plenty would have been that height. Whereas I think down here, that's actually one of the sites I really like. It's actually a volcanic basalt. So there's only about 5 to 20 centimeters of sediment on top of a kind of gravel platform. And we have, I don't know if you can see the pneumatophores growing out and around above the sediment, and the root mass is mostly above the sediment. So it's quite different. But I was hoping I would have, I think it's probably this picture right here in the middle. I'm probably actually standing within, there are probably 100 mangrove trees in that picture. Whereas this one down here at the bottom, kind of near the right where we've got somebody crouching, that one actually only has about 4 mangroves in it. So we have huge differences in locations where we might have places that have very huge, tall mangroves that have a kind of canopy width of 3 or 4 meters. And other places we have many very, very dense mangroves, particularly in our Bay of Plenty sites that might be a canopy width of 20 or 30 centimeters. So you just kind of have the one tall branch, but nothing beyond that. So just very, very different morphologies. And a lot of it, I assume, is due to both the tidal flow. So we tend to have these big tall mangroves more likely on the edge of channels, and then the very kind of dense mangroves as you get further upshore. But yeah, huge differences in particularly in the shorter mangroves, in the density and the canopy width of the shorter mangroves. Whereas the tall mangroves, when we do tend to have the tall mangroves, we'll have a lot of places where you can actually walk in amongst them. So it feels much more like a tropical mangrove environment. Whereas our short mangroves, you have a lot of places where you pretty much need to bulldoze through a lot of trees to, not bulldoze, but you have to kind of maneuver your body reasonably uncomfortably through very dense forest in order to get into sample. So, but quite different in terms of tree height. But normally we call the dwarf mangroves anything less than a meter, which I think was your original question.
Yes, so they don't represent young and versus old mangrove?
No, yeah, so we do have
okay.
Adult mangroves, and that's also a challenge in management because a lot of the councils have rules on a minimum height to be called a seedling. And so if they have a seedling removal consent, typically they can remove anything that's shorter than 80 centimeters, but it's not uncommon in the dwarf mangroves to have adult fully branched trees that are only a meter tall or that are 50 centimeters tall. And I'm trying to think, there's bound to be some in one of these pictures. And where will I find one? I'll see if I can find some good pictures. And of course I won't when I'm looking for them. But yeah, actually these would be a good idea. These are all about 60 to 80 meters tall, and those are about 10-year-old adult mangroves that are that all have seeds. So they're all actively reproducing mangroves, but it's quite variable. But it's more based on age rather than height on when the mangroves reproduce. And yeah.
one quick question. So they are all the same species of Avicennia?
Yes.
Oh,
Yeah.
okay, thank you.
Yeah. So hugely different morphologies. And so like these ones, you can see they're more kind of like tall unbranching species. And a number of these were reproductive after about 14 months. So that we did have seeds on a number of the mangroves here, and we knew their age because we actually had the photographs and the data on when they recruited as seeds and seedlings.
Thank you very much.
Hi. So really good talk. I had a question regarding the community planting experiments that you guys did. And I guess the question is, did you look at soil makeup and soil chemistry in determining where you're outplanting these mangroves, or was it just based on community direction of, hey, we think we need mangroves here? Because I have been involved in a couple aspects where if you don't, at least here in the Caribbean, if you don't pay attention to the soil community and the soil structure, it can really affect the success of your restoration.
Yeah, and I think in our case, with the community, or at least with this particular community group, it was 2 sites that they selected that they were interested in, and it was both places where they knew they were seeing seedlings. So in their case, you know, we didn't actually pick the sites, they did. But in terms of what types of metrics that we collect, we occasionally will do nutrient chemistry. Normally we do grain size and organic content, and our organic content measurements can actually get quite high. So a kind of normal sand flat environment would normally be kind of less than 2% organic content, and a lot of our mangrove sites will sit at kind of between 20 and 40% organic content. We have occasionally done metrics on dissolved sediment nutrients, so nitrogen and phosphorus, though those tend to be expensive for us as well, so we don't do those very often. And then we've also, I think the only thing that we've found is a very significant driver of whether mangroves can survive or not is the sediment oxygen levels that tend to affect the seedlings on the surface. So those early seeds and seedlings, and there we had a number of places where we checked, and it was mostly after mangrove removals. We were looking at both sediment oxygen as well as water column oxygen when the tide came in, and how quickly the oxygen was used. And mostly a lot of the sites, we had a lot of bacterial decomposition. So you were quickly seeing the water at incoming tide going anoxic that was having a bad impact on the whole estuary, not just the mangroves, but it certainly impacted on mangrove health as well. But we haven't done much else in the chemistry, mostly because our mangroves tend to be quite happy most everywhere in New Zealand. So a lot of the, if there's a lot of work on chemical pollutants around Auckland as part of their estuary monitoring, and there are very few of, I think occasionally copper and zinc in some of the areas, and PAHs occasionally are higher than New Zealand guidelines, but in typically no cases are, in Auckland being one of the more polluted urban estuaries in New Zealand. This is our Waitemata Harbor. It still typically has very low levels of any of those chemical pollutants compared to much of the rest of the world. So that we're really not on the radar where it's likely to be affecting mangrove health.
Yep, thank you. Yeah, for the next question from Miss Anggia, please.
Hello, I'm Anggia from Indonesia. I want to ask about changing perception about mangrove from 1980s. You said earlier the people from 1980s think mangrove is smelly and blocking the coast and kind of that. Because in fact in here that happening, they cannot feel the benefit of mangrove present here. They don't know, they cannot feel directly the benefit of mangrove, like uh, mangrove as the carbon sequestration, and like mangrove is a nursery ground from for fish, or to against the typhoons and tsunami, because it never happened here. So they cannot directly feel that benefit, big benefit. I think only researcher maybe can feel that benefit. So the question is, how do you think we can change the perception of the local people here, or in the other word, how to make they feel the benefits of mangrove presence here? Thank you.
Thank you. And I think it's a very good question, and I don't have the answer. But certainly in New Zealand, it's been a lot of work. It probably helps that the number of coastal storms and flooding that we've had in the last decade have been quite significant. And also one of the things we've anecdotally noticed is that in the areas where we have a lot of mangroves, we're less likely to have impacts of nutrients in our estuaries. So in a lot of the South Island estuaries, we have another big problem in New Zealand where we have a lot of dairy cows, and because the price of milk is increasing so much on the global market, we've increased the number of cows that we have on land by about 10 to 100 times, depending on where you are. And it looks like the areas like the Firth of Thames, and that area that drains into the Firth of Thames is one of the big areas for cows, but we don't have nutrient problems there because we have a big mangrove forest that's actually then capturing and retaining all the nutrients. Whereas down in Canterbury and in Southland, where we have similar issues with cows, we have big algal bloom problems that are clogging up the estuaries because we don't have those. This is my theory, that because they don't have the mangrove forests, most of the salt marsh has been cleared, that they don't have those natural nutrient traps that are helping prevent things. But I think in terms of New Zealand and getting communities on board to, I don't want to say like mangroves, I think most of it is more familiarity than them actually becoming appreciative of the values that mangroves have. I think in the kind of blue carbon world, we had a grant proposal about 5 years ago, and the feedback we got from it was we were working with an Indigenous group, and they said to us, well, why should we care? You know, we care about the trees being there. We care about, and in this case, they cared about, we have invasive Pacific oysters that would attach to mangrove roots. So they cared about the mangroves as a place where they could harvest invasive Pacific oysters. And they cared about them and being something that they saw as protecting land erosion, but they really had no interest whatsoever in blue carbon. And so, you know, how much of it is something, can we actually get communities that don't benefit, you know, directly from some of these things? So if you're not fishing, the role of mangroves as a fish nursery doesn't matter to you. If, you know, blue carbon, it's such a big combine, you know, mangroves are such a small part of a total global solution that people probably won't see what it is directly there, that keeping the mangrove forest is there. So I think in a lot of how we're looking at coastal management, it's more looking at mangroves as one thing with an entire harbor management, you know, so managing the entire catchment. And, you know, I think people learning that often how people get interested in environmental health is because they want to remove mangroves. But as they learn more and more in New Zealand, they become more interested in all the problems that are causing mangrove expansion rather than removing the mangroves themselves. But certainly it's taken, 20 years with a lot of these groups. And we, rudely occasionally say that, there's a particular age group that's post-retirement that has time to care about removing mangroves, and they're gradually becoming less and less of them. And our younger age groups that are moving in seem to potentially care a bit more about the environment, or maybe they don't at all and they're too busy doing other things. But we're certainly seeing a shift in people's preferences, and how much of it is just more general understanding of climate drivers in New Zealand and pushes for healthy biodiversity that does in New Zealand get a lot of press in the news. So, we do have a lot of public attention on biodiversity, and, New Zealand is known as this clean, green place, even though if you look deeply, it really isn't terribly different from anywhere else. We just have a very large amount of land with only 5 million people on it. So we don't, our impacts are pretty high per capita, but they're pretty low relative to the amount of area per person. So, but yes, it's very hard to get people to care about things that are intangible. And and so it's been a lot of learning and working directly with people and trying to create, the social media to do, the webinars now seem like something that we do all the time, whereas 3 years ago, what's a webinar? Who would have done that? But, trying to come up with different ways to help people learn the benefits of mangrove ecosystems is, in social media and creating, we have a lot of like little 2-pager things that are available at councils that say, this is what a mangrove does for you. But a lot of it is really working with people and not, immediately going, oh, we used to call a lot of the mangrove people mangrove terrorists.
I don't care.
Yeah, and they didn't care. But if you never talk to them, you never get to understand what they do care about and what their reasons are. And by, working with a lot of the groups, we're actually, making a lot more progress in healthier estuaries, because we have a lot of things in common. We might not have the mangrove removal aspect in common, but a lot of the other things we share are the same.
Yeah, I think I agree with you. The people may be busy with their business, right?
Mm-hmm.
Okay, thank you for the answer.
Thank you, Mbak Anggia. The next one is Pak Zulham, please, Pak Zulham.
Oops, I can't hear you.
Pak Zulham, you still mute your
Okay.
Yeah.
There you go.
Sorry. My name is Ilham. I'm from North Sumatra Province, Indonesia. A short question. What are the benefits of having monospecies of mangrove compared to Indonesia with various type of mangrove? Because I think it will be different of treatment, I mean in mangrove rehabilitation. Thank you.
Yeah, and I think, first, I'm quite jealous because we just have one species. It's no fair. And it's the same with seagrass as well. We just have this one tiny little seagrass species that the blades typically are only about 8 to 10 centimeters. So, one species of mangrove, one species of seagrass. It's not terribly interesting, but I think for me, I think the kind of zonation patterns of mangroves, and then more the challenge in restoration of mangroves. And I can't remember, I think it was Rulio that asked, someone was asking about the, kind of chemical considerations when you're planting or restoring mangroves. with Avicennia, it's very morphologically diverse and it's very tolerant of different conditions. And where you have multiple species, they often will have particular tidal heights that they're most appropriate, and freshwater flows and things that they're good at. So I think in other countries where you have multiple species, it's that much more difficult, where you actually have to consider what species goes best where. Whereas in New Zealand, we've just got the one, and it, grows like a weed. So it's typically not a hard challenge to find somewhere that mangroves can go. They have transplanted mangroves, I think, to Wellington and the southwest coast of the North Island as well. So once the mangroves are past the seedling stage, they're pretty frost tolerant. So they can, again, live, we find them in gravelly sediments. We find them on these volcanic basalts. They don't need a lot of sediment, but our Avicennia is capable of living just about everywhere, it seems. But I think, that's a challenge in mangrove restoration elsewhere, is understanding the species sensitivities to sediments and hydrodynamic conditions and freshwater flows as well.
Pak Zulham?
Thank you.
Thank you, Pak Zulham. The next question from Pak Sumiran.
Thank you, Pak Suyadi, and thank you very much for sharing your knowledge and your research experience, Dr. Carolyne. My name is Umir, and I'm from Indonesia, currently stay in East Kalimantan. And my question is, when we want to restore the mangrove area, should we consider the type of the mangrove treatment in the past that caused the mangrove degraded, like probably in the past the mangrove were excavated or manual removal, or probably affected by the natural damage. So, so in the restoration program, when we want to replanting the mangrove in the area, should we consider those type of the mangrove treatment, before we want to planting the mangrove? Thank you.
And I think, potentially, we've done similar things with seagrass in New Zealand, where we'll know where seagrass was, and then we plant it there again and trying to restore it, and it doesn't grow. And we're often challenged to figure out why. A lot of the things that you need to look at are what has changed and what caused the original die-off. So I know we've also had some natural die-offs of mangroves as well. As well as some human-induced ones. So, we've had a couple places where communities have dropped rotenone, so to take all the oxygen out. And in those places, we don't see, we can't restore the mangroves because it's just that kind of long-term chemical. They tend to be very small areas. So, I mean, the one I'm thinking of is maybe like 10 by 10 meters that are impacted by rotenone from one of the illegal removals. But it's looking at those, is there something that's a persistent chemical that somebody used in New Zealand with mangroves in regrowing? We look at whether there is the below ground biomass still there. So do you actually have somewhere that the seedlings can penetrate and get in? We tend to have very thick root mass that's left behind, and it persists for about 10 years or more, so that while it's still there, it's often a struggle for the mangrove seedlings to actually be able to attach and get into the mud. But other things that are really common problems, and this is just in New Zealand, but might be elsewhere, is to look at the changes in flow conditions. And that's typically been our biggest problem with seagrass restoration, is that the flow conditions would have changed, so that somewhere, whether it's dredging in a harbor or something, but that we suddenly have much higher flows, so the seedlings can't actually stay attached anymore. And so that's, I know that's been our experience of the places that are more likely to get washed out because something's changed to make the wave exposure more, and it's usually dredging or removal of a sandbank or some other protection that was there that helped to keep seedling erosion from happening.
Okay, thank you. Maybe one more question, may I?
Mhm.
Yeah. My second question may be related to Pak Zulham questions. So I also wondering why in your area, in New Zealand area, just have one mangrove species, Avicennia. Have you or your institution or your local government tried to planting another species of the mangrove in there, like rhizopore maybe? Thank you.
Yeah, and it's interesting because I know, some parts of Australia just have, mostly Avicennia, but you do have the Rhizopora and other species in temperate climates. And I would assume the reason we don't have it would have been just the dispersal barriers that were that far from the country, from Australia, which would be the main source. And I know looking at, we did a study of where mangroves were across all the Pacific islands as well. And depending on what island you are, it's almost like the distance to Indonesia, you find more species the closer you are to Indonesia, but then less and less and less species as you get further and further away. Yeah, so I would assume in New Zealand, it's just that colonization problem that we never had any, other species make it here, or that they didn't survive when they did make it here. I haven't heard of any other, because Rhizopore is the one that has like the very long, thin seed, I think. And I don't know how well they last in the ocean. Some of the studies we've done looking at Avicennia seeds, that they're still able to set and become seedlings after kind of 3 weeks or so floating around. So they do have a pretty long viability, so that, it is possible that they could be in ocean currents for 3 weeks or more and still land and colonize. And I think that's why they're one of the most common species down here, and also on the Pacific, and a lot of our Pacific neighbors. But yeah, no one's ever tried that I'm aware of planting another species in New Zealand. That would be interesting to see. Yeah, and certainly I know, our closest neighbors, many of them have more than one. So yeah.
Yeah, I think that's interesting to try. Thank you, Dr. Carolyne.
Okay, thank you, Pak Sumiran. And the next question from Mr. Sigit Febrianto. Okay, thank you, Pak Suyadi. Good morning, Dr. Carolyne. I have a question. Based on your experience, how big is the effect or impact of the marine debris on the growth of mangrove roots?
Sorry, can you say that one more time? How big is the impact of what?
The impact marine debris.
That's on the word I'm not getting.
Of mangrove roots. Because in Indonesia, many marine debris like plastic on shore.
Oh, marine debris. Yes,
marine
sorry.
Debris.
Thank you.
Thank you.
Mm-hmm. It's an interesting question. I know we have a lot of sites in Auckland where it's, mangroves are seen as low quality habitat and are actually not just kind of the small marine debris, but you'll see, grocery carts and, bags of rubbish and things dropped in them. And that was actually the site that we worked on with the community group. That was part of their goal, was to create the area as a recreational reserve so that people would stop dumping rubbish. But certainly, I would have seen enough kind of large rubbish. I don't think anyone has done anything looking at the effects of the smaller plastics. on mangrove growth. I know people have looked at their prevalence in New Zealand, and it's, you know, like everywhere in the world, it's quite high, the amount of those microplastics and things, but I'm not aware anybody's actually looked in New Zealand at their impacts on growth. So I don't know.
Okay, thank you.
Okay, thank you, Mr. Sigit. And the next question from Ms. Narjas.
Hi, good morning, good afternoon, everyone.
Morning.
First of all, thank you, Dr. Carolyne, for this really interesting talk. And what's interesting is that similar to New Zealand, where I'm from Bahrain. Bahrain is also home to one species of mangrove, which is also Abyssinia marina. But we're on the other end of the spectrum where they only occur naturally in 2 sites, and we're trying to expand the cover of mangroves in the country. And the people's perception towards mangroves is that they're not really aware of its existence unless they're directly involved with mangroves, as in maybe they're environmentalists or researchers or fishermen. So they're not really aware of the existence of it or their significance. So, and we do have a mangrove planting project going on, and it's been quite successful, but actual research and baseline data is quite lacking. So I'm not sure if this is relevant, but I was wondering, I'm considering all of this, what do you think would be a good starting point in terms of research for mangrove restoration?
Yeah, so I think, you know, the important part is knowing where they are now. And if you can get an idea on almost like why they aren't anywhere else. So what are the suitable conditions in the places that they are found? And that can help you determine what other areas have those same conditions that you might be able to plant or expand them.
We do have quite a rough idea. It's because of the salinity. I mean, Bahrain is quite a harsh environment. It's dry and arid, and the salinity of the soil is quite high. So it's very apparent, but how do we work around that?
Yeah, and that might be the big limiting factor where, yeah, and that's one, you know, another lovely thing that we don't have to deal with in New Zealand or never have thought about is the soil salinity. You know, I know at some point in time, I remember we measured it early on and we never got any differences in measurement. So we stopped bothering because it was, you know, never an issue, but that we do have good tidal flows and we do have good mixing in most of our estuaries so that, you know, we don't have, except in big flood events, that's about the only time that we'll actually see a lot of stratification. But that's mostly that we'll have these huge freshwater flood events where we'll have that kind of top layer of the saltwater and the freshwater on, do I have that right? Or no, the freshwater layer on the top floating out.
We do have that problem. The water is quite stagnant, so there's no mixing.
But it's, you know, we do have these wonderful, well-mixed estuaries, so you tend not to see a lot of that stagnant water. And where we have seen a few of the natural die-offs is where we've had changes in the flow in Tidal Creek where areas have been blocked off and they've become stagnant. So, and I would guess we've always assumed it was just flow, but it's probably that increasing salinity and lack of oxygenated waters and lack of flow that probably impacts. So I would guess that's probably the big driver of where they are now. And potentially where they could be expanded to. And some things, I think that's the challenge also is, you know, climate is changing. Are places getting drier? Do we have changes where we're getting, you know, in New Zealand where our climate is changing, we're an exceedingly rainy place most of the time, but where the climate changes are happening is we're getting more big events. So our total rainfall, instead of being over 50 or 100 rainfall events, is down to about 5 big rainfall events a year. So you get these huge big rainfall events and all the rain comes at once and then goes downstream and goes into the ocean, but you're not having that kind of constant rainfall conditions like we would have had 20 years ago. And that's the biggest change more on the East Coast, whereas the West Coast are just getting more and more rain and more soil erosion. But yeah, I would say, you know, understanding if it's really the salinity that's the big driver of where they can live, and are there any other locations that might have suitable salinity, that might be the bigger driver of where restoration might work. or expanding more mangrove areas.
That was very helpful. Thank you.
Thank you very much, Ms. Narjas. Any more questions? We have around 10 more minutes. Okay, no one. If no one, I'm going to ask.
May I?
Okay.
Sorry.
Yep.
Yeah. I interested in blue carbon study, Dr. Carolyne. So I interest when you show the carbon profile in the sediment. So it's very quite fluctuated, I think, in your area, the carbon of the depth. So I just wondering why the carbon is so fluctuated with depth or so varied with depth. Is there because of the in New Zealand has 4 seasons? My question is actually is the seasonal variation in New Zealand also affect to the carbon stock in the soil?
Good question. I would guess no, but that's probably because we've never looked. Most of the time when we've collected the sediment cores, we've done that in February, and I'm trying to think if we've ever done it where we've collected them multiple times in a year to look at the sediment stocks. But I think most of the variability that we see is actually that we we'll have enough cores that we've done kind of like to a meter depth, and then we use the kind of radiocarbon dating so that we can see the all the big pulses in the 40s and 50s that you can actually date things. But the bigger challenge we have is usually the top 20 centimeters or so, where we have a lot of bioturbation. So we'll have animals moving around, and that actually makes it really difficult. And that's also where you'll most of the root biomass is in the top 20 centimeters. So there's that really kind of obvious peak if you go below ground in the carbon biomass where that main root biomass is. But then as you go down deeper, there are a number of things that we'd seen. Some would be places where you see kind of older trees that are, building on top of trees, so that, again, we've got this sediment deposition that's probably on average a couple millimeters a year in every, I think that would have been pre-European colonization. And then some locations it's maybe up to, 20 millimeters or more in a year of sediment deposition. So we have places where we have lots and lots of sediment coming in, and trying to detangle the sediment, how much sediment we have landing at a site, and then we've got the animals bioturbating and moving sediment around. But then we also, as you look deeper in the cores, some places we'll see weird blips, and it's where previously there was a shellfish site. So that often, 50 years ago where mangroves are now, there might have been a bed of shellfish. And so we'll have a lot of shells within the sediment signal that often pop up, showing a very strong carbon signal as well. And that tends to be a lot of our deeper cores show that change in habitats over time that we often have no evidence to know what really was there. And this is what my colleague Andrew Swales does a lot of. Then he'll do the whole they do all the pollen sampling and all the shell sampling and all the geology of the cores to look at change in habitat and how that affects what the carbon is within the core at different depths. But yeah, it's definitely hard. And I know one of the questions we want to ask is, like a question for a resource consent. People would ask us in Auckland, they want to put a 1990 limit. So any mangrove from 1990 onward, they want to say is less important than older trees. And so we've been asked, so what is, how much does blue carbon increase with age? And it's really hard to separate out the kind of hydrodynamic variability between sites with the erosion of the animals at the sites, with when the mangroves colonized, and try and give that kind of last 20 years of carbon deposition and give kind of a rate of increase in sediment carbon per year as mangrove stands colonized. We want to do it, but we haven't successfully done that yet.
Oh, yeah. Thank you, Dr. Carolyne.
Thank you very much. Maybe we have 3 more minutes, if one question, maybe, if any. No? Okay. I have a very quick question for you, Carolyne. Are the community still continue to remove the mangrove now, or stop after the mangrove management bill?
It depends where. So it's interesting that one of the women who is very much pushing for that bill in Parliament, she's also one of our few mayors who's I don't want to say an anti-vaxxer, let's not get political, but she's in the news again right now because she's unwilling to get vaccinated as well. So she does have a interesting set of political views in addition. But I think a lot of in Whangamata, where the legal removals were very successful and we had very good buy-in, we're probably down to a few dozen members of that community that still don't like mangroves and still do illegal removals. However, most of them are concentrating their efforts on seedling removal. So I know one of the guys who we know he did illegal removals, even though he was never caught doing them. He goes out and he has like a little weed whacker that looks like a golf club, and they do have a consent to remove seedlings, and he is putting more of his effort on his daily walk. He goes out and removes seedlings. In the Bay of Plenty, that's where that big mechanical mulching that was about 100 hectares. Most of the community groups have been spending their resources on hiring seedling removal. And that council has basically completely turned against doing any additional removals. But due to kind of the nature of all the sediments and, likely the chemistry involved in all the very degraded sediments, we haven't really seen any further expansion. We'll see seedlings colonize, but they tend not to survive very well in the mulch sediment areas. So they haven't had huge issues with any further expansion there. And that council, they actually put a lot of money. I should have put a picture of this up there. They were, I'm not sure if everyone knows what a jet boat is, but they're, kind of these boats that, and then they also had like a hovercraft, but they were trying to design a hovercraft mower that could fly along and remove all the seedlings. And so they actually got a consent to do that, but that's kind of the southernmost council was working on mostly seedling, dealing with seedling expansion, but they'd already, removed a fairly large proportion of their mangroves. So that was a complete change. Whereas on, it really depends on which council and what their priorities are now, but we're getting a lot more consistency in the regional policy statements, which then when people apply for consents to remove mangroves, that they're now consistent across all of the different, all of the 4 councils, or certainly more consistent than they were before. And then the select committee, the parliament bill that it didn't actually got declined. They accepted it, but they put more restrictive conditions on it than had ever been in place before for mangroves. So in the changes that were recommended, then they just threw it away because they realized it was making it harder rather than easier. But yeah, but we still have a few people wanting to remove mangroves, but I think it's becoming smaller and smaller. And it's more, how do we, after we had a lot of pushback and a lot of changing the language to make mangrove removals more restrictive, now it's how do we cope with the, gas and electric companies and flood removal, and our Department of Conservation managing pathways and boardwalks, that they actually need mangrove consent conditions to maintain infrastructure and things.
Okay, thank you very much. The time is up now. Thank you very much, Carolyne. It's very interesting to know about the temperate mangrove, especially in New Zealand. Although, as Carolyne mentioned before, although New Zealand has only one species of mangrove there and distributed only in the North Islands, but the area is expand, and a lot of as we know from the presentation, that the evolution of the community perception to the mangrove and then mangrove as well. And actually in Indonesia we have a similar challenges. For example, the sedimentation as well associated with the land cover change and mangrove deforestation and degradation and plastic pollutions as well. So maybe in the future, all of us, we can develop a collaboration to do research or anything there. Thank you very much, Carolyne, and
thank you, and nice to see you again.
Yeah, nah, hopefully. see you later. And then the time I give to Mr. Yunus. Thank you.
Yeah.
Thank you.
Well, thank you very much, Dr. Suyati, as the moderator, and also Dr. Carolyne Lundquist for the great presentation. And now we are having a photo session before moving to the next, next explanation by next speaker, right? So, a second, please. Dr. Carolyne and Dr. Suyati and all participants. We are having a photo session, and I have 2 pages on my laptop, right? So firstly, for page 1, all right? I'm counting 1, 2, 3. Second, please. Okay. Next, page 2. Okay. 1, 2, 3. All right. Thank you very much, Dr. Carolyne, Dr. Suyati, and all participants. And next, Pak Safran, I think now is your session, right?
Yes.
Okay. Time is yours, Pak Safran.
Okay, thank you very much, Mr. Moderator. We're going to continue the Google Earth Engine hands-on training. I'm going to share my screen. Okay. Okay. Okay, let me check. Can you hear me clear, everyone?
Yes.
Yes.
Okay, good. Does my presentation displayed correctly?
Yes.
Okay. Let's continue from before. Today we're going to focus on how we can use Google Earth Engine to map mangroves. So actually, in previous exercise, we are currently still lacking one type of exercise. So it's actually Google Earth Asset Management. So Google Earth Engine currently having a lot of features, and one of the most important feature is the asset management, where you can import asset which is including an image. The image is in 2 type of file extension. The first one is GeoTIFF, and the next one is TF record and JSON under the JSON. And the next one is table. Now this is quite confusing because in Google Earth Engine, Tables includes geometry, feature collection, and even CSV file data. So flat tables and geometry tables are considered as a table in Google Earth Engine. So the best thing with asset is that you can reuse asset from your previous analysis, and also aside from using it from the previous analysis, you can export also the asset to your Google Drive, and also share the asset to be used by other people. Currently, you have a limit of a maximum of 250 gigabytes or 50,000 files for your storage in the image import asset in asset management. So how do we do this? First, we have to click the asset tab and then click new, and then you can upload images, GeoTIFF or TF records or tables. Shapefile should be accompanied with the .sxx and its .dbf files, or if you don't want to upload it separately, you can zip the shape files into one zip files, and then upload the zip files and let Google Earth Engine extract it and input it into the image asset. And you can also add asset by only clicking the arrow icon. So it's very easy for us to use the asset. So I'm going to show you how it's done. So here we have the asset. Okay, I'm going to create a new asset, which is a shapefile. This one. Okay. Afterward, you will see in the task button, in task tab, the asset is currently uploading and ingesting it the server into the server. So I'm going to give you several files given by Pak Wayan and in the chat box, and please do the asset upload by yourself. So I'm going to return the screen to the presentation. Okay. Okay, please import the new asset to your Google Engine Asset Management by clicking the Asset tab and then click New, upload the images, upload the shape files, and see it ingested by Google Earth Engine. Okay, has everyone completed the upload of assets? I think everyone's still processing the asset. Okay.
Can you please show me again? Please,
oh, Oh, the process. Okay.
The process, yes.
Okay, okay. I'll show you the process. Okay. Okay, so I'm going to redo the process. First, in Google Earth Engine interface, go to Asset tab. In this tab, go to Asset tab and then click New. Oh, it's better that I delete the. Oh, I'll delete it later. Click New. In Table Upload, choose Shapefiles. So, Shapefiles. It's up to you actually if you want to upload the shape files. Here you can upload source files, upload the DBF, shape, and SHX files. Rename it according to your needs, and then click upload. And check on the task tab the submitted task. Okay, if it's successful, you'll get a blue bar just like this. Okay. Okay, I'm going to pause the screen and check. Anyone has anyone completed the asset upload?
Okay, so we only upload the shape file, or we upload the other the other 3 files.
So you should upload not only the shape files. Okay, I'll show you again that what you should upload. So you should upload the CPG, DBF, HHP, and SHX files. These files. Okay.
After that, we just upload it, right, Pak Safran?
Yes, after that you can click upload and just rename the files according to your need. These files, these files that you need to upload. Okay. You can also upload image files. It's better to use GeoTIFF, especially when you have data sets that currently not available in the Google Earth Engine's data catalog. Okay, finish. Okay, everyone finish the upload process? Not yet. Oh, what kind of error? Please, do you mind sharing the screen? I'm going to stop sharing.
Sorry, Pak Safran.
Yeah, okay,
I can upload just one file.
Oh, you don't need to upload the .kpg file. It's a kpg is a QGIS projection file, so you don't need to upload the kpg file.
Okay.
So DBF, SHP, and SHX. Yes, the most important things are those 3 files. I think in the asset name should not have any spaces. Maybe you can use the example I upload in the chat box.
Okay.
Okay.
Should we be using the files you sent in the chat box or the
Yes.
files that were sent on day 3? Because just want to make sure I use the ones in the chat box. But...
I use the one in the chat box.
Okay, all right.
Okay.
Still,
How is everyone's going?
still updating.
Still updating. Okay. Okay, I will share the screen to show you how it's going to be when it is success. Please notice the right panel, and in the task tab, if you are if your task has been completed, you will get a blue bar like this. Okay. Has anyone get the blue bar yet?
I have one, just one.
Oh yeah, it's fine. It's fine. Because I have 4 because I experiment earlier.
Oh,
to show you how it's done. It's fine. 1 is enough.
Okay.
Okay, let's see the chat. Okay. Okay. Cannot click the upload. Okay. Cannot click this button, Mr. Wiwit. Oh, sorry. Can you
Yes.
sorry, Miss Wiwit. Can you share your screen? I will stop my screen
Wait.
share. Okay.
Sorry, is this Zoom session is recorded? Ongoing or not?
Yes.
Okay, I think
Yes, it is recorded.
Oh.
I think the problem is you are using the files with spaces in the file names, and Google Earth Engine doesn't allow spaces in the file names. So it's better that you use the files I sent you from the chat box.
Okay, thank you, Mr. Safran.
Yes, you are welcome. We try to do this because it's one of the most important features in Google Earth Engine, because previously we use data that are available only in Google Earth Engine, but if you have your own data set, such as having a ground truth data or other type of data, you should better use your own data instead of generating it in Google Earth Engine. And therefore, this step becomes crucial for you to be able to use Google Earth Engine to its fullest potential. Okay, I think Miss Wiwit has solved her problem. Oh, Diana, you don't get the blue results after
issue, I think.
Not yet.
Not yet. Oh, still processing.
Okay.
Ingesting.
Okay, thank you.
Okay then. Okay, nice. Okay, anyone? Okay, Dr. Chu, please share. Oh, I think you missed the files. Okay, I will try to send it again. Okay, that one. Okay. Okay, nice, Mr. Sigit. Okay, nice.
Sorry, Mr. Safran, can I ask some question?
Okay.
About the assets itself, is it really take a time, even the we know that the shape files or the DBF, it's a small files to upload. Are there, if we upload five minutes?
Okay, I think the main problem is not the upload process, but the ingesting process, because after the upload process, Google Earth Engine will try to parse your SHP file according to the schema that is provided by the Google Earth Engine. So Google Earth Engine will remap your data according to according to the format available in the server. So that's why it usually took quite some time during the ingestion process, not the upload process.
Oh, okay. So it's the ingesting from the Google Earth Engine itself, not the uploading. Okay. Thank you, Mr. Safran.
Yes, you are very welcome. And afterwards, don't forget, if you want to check on your new asset, you can go to the asset tab. I have to cancel this first. You can go to the asset tab and click refresh. Now you have the new asset right here. This is the description of your asset that you've already provided the information to. And then if you want to import it to your project, you can just click on this arrow. You can name it according to your needs. And then after you have this, you can play around with your own dataset instead of using the dataset from Google Earth Engine. So this is the process of uploading and managing assets using the Google Earth Engine interface. Okay. Okay, then I think we can continue to the next step. Let's go to Mangrove. Yay. Okay. So actually, there are a lot of references about mangrove mapping using Google Engine. I tried to summarize into 3 most common types. So actually, mangrove mapping in Google Engine, there are about five methods in mapping mangrove. The first one is using the visual interpretation. The second one using algorithm based, and then the third one using machine learning, supervised classification using machine learning, and the next one is object-based image analysis. So in this training, we're going to learn 3 out of the 4. Actually, there is another one, the unsupervised algorithm, but you have to understand your area first before you can use the unsupervised. So we're going to skip it and we'll do it for later on. So how to do it for visual interpretation? It's actually very easy, since Google Earth Engine currently equipped with high resolution satellite image that you can use to identify mangrove visually using the available views, and also use the geometry tools to mark the mangrove area, and then afterwards you can input the asset. So the only thing you need to do is you should pan your to your intended location. I think most of you has already preferred area. You can continue using that area. And then previously in the base map, you are currently using the Google Maps base map. So it's not that informative unless you want to focus to the label. But if you want to see the actual features within the Earth imageries, you can click the satellite button. And then after that, you can use the geometry tools to draw polygons of your mangroves. And then you can name your polygon accordingly, and then export it to asset and to drive using the following script. See, this is the following script. This script focus on export. So Mr. Bayu will give you the snippet, and I will show you how it's done. Okay, so we're going to delete this. We're going to delete this, and then we go to, okay, we're going to go to UK, not UK England, but UK Ujung Kulon in Banten province near Jakarta. So we go to satellite. Okay, as you can see here, we have our satellite view, and then you can see the difference between mangroves. Okay, you can see the difference between mangroves, and terrestrial vegetation. And terrestrial vegetation, as you can see here, it's quite different. And you can just use this tool, the shape tools, geometry tools. You can change the geometry name to mangrove, and then use feature collection, add property, habitat, mangrove. You can add as one mangrove, and then you can just draw the polygon delineating the mangrove areas like this. Sorry, for the sake of the training, I'm not going to be very thorough with the digitization process, and you don't have to. As long as you have a good polygon, that would be enough. It would be sufficient for the example we're going to use. Okay, you can do it just like this. If you have a lot of time later on, you can play around with these tools, the geometry tools, and digitize your own area of mangrove. And you have a new mangrove dataset. Oh, I almost forgot. Don't forget to always create a new geometry layer called area of interest, or AOI. Never forget this step. Okay, to delineate the area of interest. Don't forget the area of interest. Geometry type is geometry, but the mangrove is feature collection. Okay, this is the difference. Okay, you don't have to show the AOI polygon. It's not that important in the script. Okay, so this is the example of using visualization and geometry imports and the geometry tools. Okay, and then after that, you can upload. You can save your data to the asset, And also you can also save it to your Google Drive. Okay. There are actually two types of export in the script. I will show you later on. Okay, so after clicking run, okay, as you can see here, this script focus on calculating NDVI, and then after that, this is the important script. This script focus on exporting the image, exporting the composite image, exporting the composite image within the mangrove region, within the mangrove region. So it will only upload to your Google Drive this area, this image area, image in this area. It won't upload image in the area of interest, but it will upload the area within the mangrove region. So the first one will export the data to your Google Drive. So you can download it for maybe layout it in the Quantum GIS software. But the second one will export it into your asset. Okay, so you have two type of export. It is a good practice if you have a very big analysis. It is better that instead of just visualizing it, you can also export it to your asset. It is much better like that. And then the next script is the export for shapefile.
Mr. Safran, sorry for interrupt you
Okay.
because I see someone raise her hand.
Okay.
Patrice.
Okay.
Yeah.
Okay,
Yeah, please,
Patrice.
You can ask.
Hi, Mr. Safran. If you have a degraded area in your mangrove, should you not do you include that in your polygon or feature collection, or do you do it separately?
Okay, that is a very good question. Actually, it really depends on your analysis. If you only want to map the extent of mangrove, not the quality of mangrove, you can include the polygon, both the healthy mangroves and then the degraded one. You can include it in one polygon. But if your target is to delineate between the degraded one and the healthy mangrove, you should create at least two polygons, two feature collection. The first collection should be the healthy mangroves, and you can see we have property habitat. You can add another property. It is actually mangrove for habitat property, and then you can add status like this. And if it's currently degraded, you can type 0, or if it's healthy, you can just use value 1. So it's actually depend on your target of analysis.
Okay, thank you.
Okay, thank you very much, Beatrice. I think this is one of the most important aspect of mangrove data collection. Maybe Mr. Bayu can add to my explanation.
Sorry, I didn't get it. I still concentrate to answer the question in the chat room.
Okay.
What is the question, Mr. Safran? Sorry.
Patrice thinks that asks us whether if we include degraded mangrove in our polygon,
Yes.
or we should exclude it in our polygon, include it in the digitization process. My answer is it really depends on her target and on her target.
Yeah.
But if you happen to have any more explanation, please do enlighten us, Mr. Bayu.
Okay. I think Mr. Safran already give you a complete answer. I think this is depend on your aim of your study. If you want to study about the mangrove habitat as a whole, then you need to digitize the mangrove area, not only in the mangrove cover, but also the potential mangrove habitat, such as for the mudflat, something like that, and also as well as the, such as the new development land, because of the sedimentation process, something like that. Then you should includes those area in your study area. But if you want to only, for example, if you want only extract the vegetation cover over the mangrove area, so you only has to digitize in the mangrove cover area. You can exclude the potential habitat of your mangrove area. It depends, I think, of your aim of your study, Patrice.
Okay, thank you.
Yeah.
You are very welcome, Patrice. Nice to have Dr. Bayu here.
I'm still candidate.
Okay. Okay, let's see. Okay. The next one is export the feature collection. So the mangrove feature collection here is going to be exported to the drive using format usually used by Google Earth, KML, but also you can have your file format as SHP, like this, or CSV. It's really depend on what you are trying to achieve. I usually prefer SHP. So
sorry, Mr. Safran, someone asked you to repeat how to setting the properties and the value of the geometry objects.
Okay.
Yeah, sorry, sorry.
Okay, it's okay.
I just read in the chat room.
Okay, thank you. I think I think it's not quite clear because you have to go to the geometry imports and then go to the edit layer properties button with this gear icon. And then click it, and now you have configure geometry import window. So for mangroves, you can use as feature collection, and then you can add property like this, like habitat, the value of habitat. I define it myself. Mangroves, the value of its habitat is 1, and then you can also, just like Beatrice's example, you can add another property such as status, where the value of the status, maybe you can have good is 3, poor, and etc. So you can have also a value for the status of the mangrove. You can do as much as you want, and it really is depend on what you need for analysis. But the least one, you have to have this value, the property of habitat and the value of at least one number. Okay.
Mr. Safran, may I ask?
Okay, please do.
What difference between feature and feature collection in geometry configuration?
Okay, so actually in feature, if you only have one multi-polygon, if you only have one multi-polygon, which only have one property and one value, you're going to use feature. But if your geometry has probably more than one value in the property, it is better that you use feature collection.
Okay, thank you, sir.
Okay, you are very welcome. So the ones determining which you use, whether you use feature or feature collection, is the value of each property. If you only have one value for one property, for each property, it's better to use feature. But if you have multiple values, it's better to use feature collection. Okay. Okay.
I have a quick question here. So
okay,
if you're using feature collection and you wanted to add, as you say, multiple, how would you add another one here? Do you just draw another polygon and it would just add it to this same mangrove, or would you, like, how would you add a second or a third collection?
okay. So actually, the way to do it in Google Engine, you have to add the second on second feature collection, and then merge the both feature collection to come up with a new feature collection. So it's very tedious actually, but that's the way how you do it in Google Engine. That's why it is much better if you have a shapefile, you use the Quantum GIS to merge all of the data you require and then upload it in Google Earth Engine Asset Management. So it's much better you do it that way. Okay.
Okay. Yeah. All right.
Okay. Okay. Nice. Okay. Okay. Then, if you have understand, this one is for uploading to the Google Drive, but this one is for uploading. Oh, I'm sorry. It's supposed to be not to Drive, but to Asset. Okay. It's supposed to be like this. Please change your code accordingly. Okay, now this one is to Asset, this one is to Drive. Okay, so when you export the data to your Drive, you will have to create some parameter, and you can just use this type. You can just type your file name, yeah, file folder, and then the file format. You can choose the file format. So even though you have already specified the file format, you can change the file format after you click run on the sorry, on the task export window. Okay, so if you want to start the export process, you have to go to task. Previously, usually we have in console, you have to go to task and then go to each task and then click run and specify the task in the table.
Sorry, can you repeat again, please?
Okay, so actually usually after running the program, after running the script, after you click run, you're going to have the task tab blinking like this. And then after the task blinks, you can click on the task tab and then click on one of the tasks you would like to run. Okay, like this one, this one. This one is initiating table export for Google Drive. Okay, it will export the table to Google Drive. So it's actually exporting this one. Okay, and this one is actually exporting to the asset. Asset is actually doing the export for the script. And this one is actually exporting to the Google Drive. This one is doing the script. And this one exporting to the asset. This one actually doing this snippet. Okay, now we run the task, all of them. Okay, and the taskbar will have the process gear button. You should wait, and if it's all blue, then you have already finished exporting to the designated location, whether it's asset or drive. That's how you do the asset management using Google Search Engine, by combining the geometry tools and the script inside Google Search Engine. Okay. Okay. Everyone's completed their upload export process. Have everyone completed?
It's still running.
Okay, it's still running. Okay, it's fine, fine. Let's just continue with the presentation, so we can continue with the exercise. Now, actually, you have already managed to do a visual interpretation process in Google Search Engine. This step is important because you can also use it as a part of the machine learning workflow. So we're going to use this step again in the machine learning workflow. Now we're going to talk about mangrove indices. As you can see here, we have a lot of mangrove indices, from mangrove index developed by Winarso et al., mangrove recognition index, combined mangrove recognition index, etc., etc. So we have a lot of mangrove indices, and all of these mangrove indices can be inputted directly in Google Search Engine.
I think Mr. Safan having trouble. His connection freezing. Okay, everyone. While we are waiting for the Mr. Safan to reconnect his connection, this is one of the steps for the visual interpretation. So, if you remember, in the first day of this training, that I mentioned about how to decide whether this object is a particular object. For instance, this object over the image is the mangrove or not. You can use the several principle of the K of the interpretation. So including the texture, including the color, tone, and then the texture of the object, and also the site association, as well as the pattern of the object. And the most important thing is the last is the local knowledge of your study area. So before you conduct a visual interpretation, you have to know briefly about the information surround your study area. And if you don't have those information, you can use another data resources such as the satellite, high resolution satellite. As Mr. Safran said, that the Google Earth Engine also provide the high resolution satellite data in this platform. And how to access the high satellite resolution data? Just simply click on the base map layer and choose the satellite imagery. Okay, clear? Are you clear?
Yes.
Yeah. I think Mr. Safran still have trouble. And the visual interpretation also the key before you start the digital classification, because if you remember that the steps for the digital image classification, whether it use the machine learning or not, the first step is to make a classification scheme. The classification scheme is can be produced and can be generated if you already know about the information of your study area, and you already know about the variation of the land cover over there, or the habitat over there, something like that. And then after you make a classification scheme, then you will ready to defining the training area or training pixel, as you have practice in the second day. Yes, I think in the second day. And then how to defining your training area. You can use your skill to interpret an object based on the several key interpretation, such I mentioned you before, the color, the tone, and then the texture, pattern, and the local knowledge, kind of like this. Okay, do you have any question, participants? While we are still waiting for Mr. Safran.
Mr. Bayu, I want to ask.
Yes, please, Mr. Jamal.
Yes, it's okay to use a mosaic image to do classification, since maybe each image have different condition.
Different condition, yeah.
Yes.
Yeah. I think it's more wise if you do another steps before you use the mosaic image. If you remember yesterday, when you want to combine several images to generate the cloud free image, you use many images from the different time acquisition, right? It is the same process with the mosaicing process. And then how to make the color balance between one scene and another scene. The first, you can use the several statistic parameter, such as the mean value, median value, or sometimes we use also the maximum value. So that is the process. So which one better? It's depend on the weather condition and also the environment condition of your study area. But the most case, I use maybe several. Our college also use the median parameter for defining or for setting up the color balancing between the first image and other scene image, kind of like that, Mr. Jamaluddin. So you have to do the color balancing.
Thank you, sir.
Yeah. Okay. And then participant, if you still have other questions. Okay, I think Mr. Safran already with us. Okay,
I'm sorry, everyone.
Yeah.
I'm sorry, Mr. Bayu. I was bounced out of Zoom, even though my internet connection was fine. I don't know what happened.
Yeah, please.
So I'm going to reshare.
Sometimes happen also.
Yeah, this is what happened when you do it online. There's so many things that we can't understand what happened.
Yeah, that's common problem.
Okay, so we will continue to the mangrove indices. So we have a lot of mangrove indices, as I have said before, and these indices can be inputted directly in Google Earth Engine using expressions. So image expression is one of the most useful tools in Google Earth Engine because you can directly input formula. Previously, I forgot. I think it's Mr. Sigit or anyone who asked me whether it is possible to input the Lizenga band transformation. You can use image expression, and any formula you can input it over here, even though you'll be needing a lot of learning on how to input the formula in Google Earth Engine. So we're going to use mangrove vegetation index. Mr. Bayu used this in his dissertation, in his research related to mangroves. So actually, the focus of mangrove vegetation index, this index focus on the different values between the shortwave infrared and the green values for mangrove that is considered smaller than the difference observed in the same spectral region of the sparse to dense and very dense terrestrial vegetation. So this index is to discriminate mangroves with terrestrial vegetation and also with water. So therefore, very dense vegetation pixel commonly have high shortwave infrared values, making it separable from the mangrove pixel. So this is the main logic between the use of mangrove vegetation index. So it is important that you just don't use an index, but use the knowledge and science behind the index to understand and use it in Google Earth Engine. If you don't do that, Google Earth Engine will only become a useless tool that cannot analyze what you need for your research and management program. So this is I provide you with the link for the publication, and you can use this link later on to study more related to the mangrove vegetation index. So this is the workflow for semi-automatic mangrove detection. So the first one you need to do when facing a problem or you want to answer a scientific question is the first thing you do. You have to create filters. You have to create. You have to create the workflow first. So you have to create the workflow and break down the steps using Earth Engine steps. Earth Engine step, not only breaking down the step using a regular GIS and remote sensing step, but you custom your steps according to the ability of Google Earth Engine. So the first one, we're going to create a filter and then open Landsat 8 surface reflectance, create a cloud-free mosaic composite, mask it by threshold, calculate mangrove vegetation index, create the free mosaic composite, we mask it, and then calculate mangrove vegetation index, and then we mask it using the threshold. So this is how you do semi-automatic mangrove detection using MVI. Okay. So the first thing you need to do is to create filters. Use the geometry tools you already practiced before and draw a new geometry on your area of interest and name it AOI. And then you copy and paste the following script, this one, the step 1 script. And what this script do is you center the map on area of interest and then create a boundary. This boundary is actually a box of maximum and minimum that will delineate area where the image collection will be gathered will be collected, where the image collection will be collected. Unlike if we only focus on the object, they usually, sometimes we can get areas where there is no satellite images available, because it is on a different scene. So if we use bounds, we can make sure that all of the required image are there within the boundary of the scene. Okay, and the next part, we construct the start and end dates. So we have the start and finish variable, and this is the temporal filter. So if you want to map mangrove in 2017 only, you can use the date for 2017 and so on. Okay, so please copy, paste, and paste the script, and then we can continue to the step 2. Step 1, we don't have any output for step 1. Okay. Okay, everyone has already completed the first step.
So do you want us to run the script at this point, or just paste it in?
Not yet.
Okay, all right. Just want to make sure.
Okay, not yet. But at least you have to create the geometry. Have you guys create the geometry?
May I share my screen?
Okay, please do.
Yeah, please, Mr. Jamal. Okay, Mr. Safran would like to display off your screen.
Yes, I think I will stop my screen.
Yeah.
Okay.
There is an error in my script.
Line 34. I think
what's going on?
This is the previous. I think this is the previous exercise.
The first.
Yeah, the previous exercise.
Yeah.
I think you then you have to delete the S from the assets. Yes, and then try to click run. Okay.
Run, I think.
Okay, nice.
Okay.
Nice. Okay, now
And then what steps?
We go to the first step. I'm going to share the screen.
Okay.
Okay, go to this step. Okay, copy and paste step 1. Copy and paste step 1. So it is better. It's actually better you create a new. It is better you create a new file to differentiate with your other exercise. Create a new file, phrase file, and then create a new geometry tools. You create new geometry.
We have someone raising his hand.
Okay.
Mr. Rulio, please.
Okay, please, Mr. Rulio.
He may have been answering. I was going to ask him if we can use the same area of interest that we had from before, but I gather that you're saying that we shouldn't.
Yeah, it's better that we create this from scratch, because it is better that in each exercise you're going to have a fresh portfolio, and then after that, after this training, you can go from one files to the other using these new files.
Okay.
Okay. Okay, have everyone completed the first step?
Yes.
Okay, complete the first step, and then we continue to the second step. So the second step, we open the Landsat 8 surface reflectance. Now the difference with the script you have before, this Landsat 8 surface reflectance, we filter it with bounds of area of interest and start and finish. So we don't put the date inside the surface reflectance calling, the surface reflectance at 8 SR call, but we put it in the variable, and then we call this variable inside the filter date. So this is one of the tips that we want the participant to understand, that if you do a lot of analysis, you just need to replace the variable at the front, the year, the year and the date, and can you just run it again? You don't have to play with the other snippet. So you only change one or two variables, and then you can do the same analysis to your other data set. This is one of the best practice that you can use. Okay, copy and paste this step 2. You don't need to click run right now, because there will be no output given by Google Earth Engine yet. Okay, everyone completed copy pasting the Landsat 8 surface reflectance data? Okay, let's continue to the third step. Now, the third step, we create a mosaic composite of the Landsat 8 data. So this is you have already familiar. And as you can see here, Mr. Bayu previously explained that how you can play around with the quality of the image composite. So in the VAR composite, we have a reducer here. We have a reducer using the median reducer. So this is how you can play around with the quality of the image composite based on your analysis. So you can have median, you can have minimum, or you can have maximum. And it really depends on your analysis. Sometimes, in my case, I've used percentile, the 25% percentile for the reducer. So it's really up to you. But the most commonly used is the median. So it's really up to you. And the second one is the visualization parameters. Please do notice that when you use a reducer, the band name will be added a name. The band name will add the name of the reducer behind the band name. So in the previous example, we only use band 4, band 3, and band 2. But since we're using a reducer, the median reducer, we will use band 4 underscore median, band 3 underscore median, and band 2 underscore median. So please notice the difference between this script and your previous scripts. And the last one is map the layer into the map viewer using the visualization parameters mentioned above. Now you can copy paste the script and then click run. Okay, I see there are 9 chats over here. Okay, okay, no. Is there anyone raising their hands? No? Okay. Okay, I think have everyone successfully displayed a new composite layer in their area of interest? Okay.
Show your screen, Mr. Safran, so you can see your result.
Okay. Okay, let's. Okay, I'm going to pause it for a while. Okay, I think this one should be it. Okay, this is UK, Ujung Kulon. Okay.
No, we cannot see your browser.
Oh, okay. I will. Oh no, sorry. Okay, I will try to share the screen. Okay, now as you can see here, this is my Landsat 8 composite. This is my Landsat 8 composite in my area of interest. Okay. As you can see here, the result. Okay. Now, after running up to step 3, has anyone experienced any problem? Okay, I'm going to pause my screen and check. Is there anyone having any type of problem? Okay, I think you're doing good up until to this exercise. Let's go to step 4. Now, step 4, masking before analysis. So the next thing we do, we have to mask the data, the Landsat data for 2 things. So we're going to use the pixel from SRTM. As you know that mangroves only lives in lowland area. So you cannot find mangrove in elevation of 1,000 meter above sea level. But you can only find it in the lowland area, especially in coastal area. So for masking, you can use a pixel below 50 meter. So you have to mask pixel above 50 meter. Okay. And sometimes if your area is very flat, you can even lower the threshold in order for you to have a better mask, to have more discrimination between terrestrial and mangrove, terrestrial vegetation and mangrove. Okay, please copy and paste the script at the bottom of the previous script, and then click run.
I have a question.
Okay, please do.
So in terms of using a smaller number for elevation, how much is that dependent on the accuracy of the layer that's going to be calculated in elevation? Because I think that you could use something like maybe 20 meters, right? But then if the pixel size or the layer doesn't actually cover 20 meters, it could then be a problem. So what is the minimum number that is actually advisable here?
Actually, it's really depend on your data set. If we use currently I'm using the SRTM data set. So it's actually it is actually have a 30-meter resolution with, okay, maybe let's just explore the data set. Okay, and we can find out. Okay, so this is the data set for SRTM. So we have the description. This is the bands. Okay, the bands minimum 400, maximum pixel size 90 meters. So this one's using a pixel size of 90 meters. Actually, there is no. Actually, the most important thing in using them usually is vertical accuracy. So actually, sadly, we don't have that information here, vertical accuracy. So if you have vertical accuracy value, that is the minimum unit that you need to use. So if we assume that this based on the band information, that if we assume that the pixel size is 90 meters, we assume that the vertical accuracy is around that value, and therefore we can use the value between 50 to 90 meters, something like that. It is much better if you have the vertical accuracy value in order for you to decide how much threshold required to mask areas that is actually considered as terrestrial vegetation area.
Okay, thank you.
Okay, you are welcome. Okay, so we're going to copy paste the step 4, and I'm going to explain to you what it do. Okay, and we can click run. Now in step 4, I've already told you about the SRTM dataset that we're going to use to mask areas. Okay, so we have the new composite. Okay, the new composite, and then we also use the Hansen image to global forest change image, and mask water area to create a land composite. Okay. Okay, so therefore we have a new variable called land composite. Okay, we can continue our presentation. If you haven't encountered any problem. Okay. Now, this is the most important part. Okay.
Before you hit that, should there be a visible difference between the output that we're seeing for step 3 and step 4?
Yes, the difference is in step 3, you still have the water pixels. In step 4, you should not have the water pixel.
Okay.
Now in step 5, this is the most important part. This is how you do modeling in Google Earth Engine. You compute the MVI, the mangrove vegetation index, using an expression. So this is how you do it. The first one, you declare the MVI. and you calculate the composite using this expression, near green and swir green. Okay.
Sorry.
Yes.
Yeah. So I think even though I've run it, I think I still do have the water pixels.
Oh, how come? Please share your screen. I will stop my screen. Oh, you still have the.
Right. So even if this is the area of interest here, but I'm still seeing the water pixels.
Oh, because please activate the layer. Activate the layer. Activate the layer. You actually have 2 layers. Visualize only the composite, not the Landsat 8 composite. No,
Okay.
the other
Thank
one.
you.
The other one. Nah,
Yes.
okay.
Thank
have
you.
the new layer.
Thank you.
Okay.
All right.
I see that in the chat that everyone experienced the same problem. That's how you.
Anila won't share their screen.
Okay, okay. Please, Anila, please do.
If I remember, it looks like that.
Okay.
Already masked.
Yes, it's already masked. I think it's already correct.
Okay.
Okay, the script is already correct.
Oh, okay.
Okay. Okay.
Mr. Safran, may I ask?
Yes, please.
Is there other data to masking beside Hansen?
Oh, actually there are a lot of not only a lot of. You can even generate your own data. So if you think that Hansen et al. Data is not sufficient, you can even create your own masking using NDWI, such as the water index, using the water index. In previous exercise, we already learned how to calculate the water index. You can use it and then use the lower than and greater than statement. The operator lower than and greater than operator, as shown in this in the fourth step. In the fourth step, you have var elevation, SRTM select elevation, which is the band elevation, mask SRTM composite lower than 50. So we mask the composite where, oh, I think I made a mistake. It's not supposed to be composite. It's supposed to be SRTM. So please run the script again. So you should have something like this. Can you see my screen? Everyone?
Yes.
Can you see my screen?
Yes.
As you can see here, areas above 50 meters are masked. So areas above 50 meters are masked. So they won't interfere with our analysis. Oh, so it's supposed to be this. So this is the I'm going to copy and paste this in the chat for you to create a new data, a new land composite image. Okay, the final product is this one, the one with the area mask, area greater than 50-meter mask. Okay. Okay, if you sorry for the mistake, and let's continue to the presentation. Okay, the masking. Okay, the next one is calculate MVI. So this step to compute the MVI using expression and rename it to MVI, and then after that display MVI. Now this is important that you also display the MVI layer, because I want you to play around with the visualization in order for you to get the threshold, according to your area. So let's play around with the result a bit. Now we have a new layer called MVI, and then in the MVI layer you should do this from custom to stretch to 100%, so you know the extent value of each of the image, and then you want to experiment with the threshold by clicking custom again. Now clicking custom again and use value like 0 or maybe like 4 to 20. So in each area they have actually different range. You have to experiment with your own area. In Indonesia, there Balaloy have already created a research, already made their research, and they come out that the range is between 4.5 to 20. 4.5 to 20. And how you know that your threshold is good is by examining the pixel. If you have by examining the pixel visually, if you have a gradation of black to white to gray to white, then this means that your threshold has good and can discriminate between mangrove vegetation and terrestrial vegetation. So if you only have like this, like all black, see, this one's good, but it also includes the other area. You still have gradation in other area. This is not a good example. So I think it was -90, the minimum value, -90 minimum value. Okay, you have this. If you have this gradation, it's not really good because it can't distinguish between your mangrove area and terrestrial area. So the visualization is important to examine whether your threshold is already able to discriminate between terrestrial and mangrove areas. I think in both. Okay, we stretch to 100%. See, if we stretch to 100%, we cannot distinguish the area, the mangrove area properly. Therefore, we have to play around with the visualization over here. Okay. Okay. Now, if you have completed the step 5, we can continue to the next step. Now, this is what I've been talking about. So we have to tweak these values accordingly. So copy and paste the script to your code editor at the bottom of your code editor. And then, sorry. Okay. Anyone want to unmute themselves? Okay, no, then we can continue to the...
Sorry, Mr. Safran,
yes.
I want to ask, so on the step 5 before,
step 5, okay.
I just want to ask that the distinguished color between the terrestrial and the mangrove in the coastal area has been must be like you said before and visualize. It must be different, like the pixel in white and black is visible and the terrestrial is different color. It's like that.
The most important thing is that you can see the gradation between within the mangrove areas. That is the most important thing. If you can see the gradation within the mangrove area, then your threshold is good. But if you see the gradation expanding outside the mangrove area, so the gradation expanding outside the mangrove area, then you have to lower or upper the threshold.
Okay.
You can have to rise or lower the threshold. So this is the rule of thumb. Okay.
Okay, thank you.
So you're going to need a very good monitor to do this.
Yes. Okay.
Yes.
Thank you, Mr. Safran.
Yes, you are welcome. Okay, anyone has already completed step 6?
Excuse me, Mr. Safran.
Okay, Narjas, please.
I'm trying to play around with the MVI, but it's completely black, and I'm not sure how.
Oh,
I'm
please.
supposed to.
Please share your screen.
Okay, 1 minute, please.
Okay, I'll stop the share screen.
Just a moment. Yeah, there it is. Can you see my screen?
Yes, clear. Okay.
Yeah. So no matter how much I play with it, it's still completely black. This is the area.
Okay. Actually, it might be possible that your area's mangrove range is outside that range. So, do you mind you go click on custom?
Yeah.
Yes, click custom and stretch 100%. Okay, click apply. As you can see here, your area's data ranges in hundreds.
Oh yeah.
Yes, it's very different. You have a very different threshold. So you have to play around with that threshold. Maybe it is better that you use the inspector and click one of the area that you know that it is a mangrove area.
This one's definitely one.
Okay, and then go to MVI.
Where?
In the inspector, scroll the screen until you find the MVI.
Yeah.
Yes. So you have 4 as the base. So you need to find the maximum number.
So I'm not really understanding. So this is 4, and then what do you do with this number?
Yeah, in this number shows that mangroves, the area you click have an MVI value of 4, and you are certain that this value is mangrove. Okay, and then you put it in the range that the minimum value is 4. Go to stretch 100% and back take it back to custom.
Okay. Custom. So I
Okay,
can edit it.
custom.
Right?
Okay, edit the range to 4.
Here, maybe let's say
Okay, maybe you can try maybe 50.
50? Okay.
50, okay, and apply. Okay. No, you still haven't seen the gradation. Oh, I think you should raise again the range.
Higher than this, let's say 100 maybe.
100, okay. Let's try. No.
I'm gonna go really high.
I think the maximum number is 190, I guess. Okay, let's try 190.
Could the location be a problem?
Maybe I think we can come back to this later.
No problem, it's fine.
Come back to this later because I think in this area you have found that the threshold of MVI is very different from the threshold that usually found in Southeast Asia. So this is going to be your new journal article.
Oh, but this isn't in Southeast Asia. This is in the Middle East.
Yeah, I know, because since MVI research has been done, a lot has been done in Southeast Asia.
Yeah.
You can use this as your new finding in the Middle East, and you can say that in Middle East we have different threshold.
Okay, okay.
So we're going to wait for your new journal article, okay?
Hopefully, yeah.
Sorry,
Okay.
Narjes.
Yeah.
There is the geometry layer over there. There is the mangrove area or mangrove area.
Mangrove area, yeah.
I see. Okay, okay. Just will you use your inspector cursor to see what's value over the mangrove area?
Sorry, I can't hear you.
Click on the inspector. Click on the inspector.
Inspector, yeah.
And then click on your screen map. Over the, yeah. Wait a second. 3.0. Okay. I think your mangrove area is quietly dry.
Yeah, it's dry.
Yeah. Okay.
So
it's
yeah, very different from Southeast
You're
Asia.
finding
Yeah,
it.
Mr. Professor Bayu. Yeah,
you're finding.
Okay, this is interesting.
Okay. After this, we can expect a new publication from Narjes. So
Hopefully.
don't forget to email us about your new finding.
Of course. Hopefully. Thank you.
And we can continue with the presentation and the exercise. Maybe Narjes can switch. Maybe Narjes can switch to other area first.
Okay.
Before doing your research in the Middle East.
Okay, so for this case, I'll switch to another area, and then I'll keep up with you with the YouTube.
Okay.
Okay. Thank you very much.
Okay,
I have a quick question.
Okay, please do.
Yeah. So if I wanted to do, if I draw a polygon around a known area of mangrove, I can look in the inspector and see what that signal is, and maybe that could actually help me. Okay. All right. Thank you.
Yes, you are correct. That is the basic principle of using threshold. Okay, I'm going to display my screen again. So this is the thresholding process. So you have to change the threshold accordingly. So this is the threshold for Southeast Asia. Southeast Asia, maybe in your area you have a different threshold due to the condition of mangrove irrigation, and you can modify the threshold accordingly, according to your area. Okay, copy paste the step 6 threshold while I'll explain what this do. First, it will declare a variable of the lower threshold and the upper threshold. So the lower threshold and the upper threshold. And then afterward, you're going to create a new variable based on the threshold. So we're going to have in the threshold, we're going to have 3 values, 0, 1, and 2. Okay, 0, 1, and 2. Where the 2 is, where the 2 in the new image is actually the mangrove. It's actually the positively mangrove, while 1, while 0 means it's almost impossible to be mangrove, while 1 is possible to be mangrove. Okay. Now, and then after that, we mask the layer, only taking layers that is with high possibility of mangroves, which is equal to 2. Okay, and then we mask it and only this and only take the mangrove area, and rename the new band as mangrove. And then finally, we display the final product as it is. Okay. So I'm going to show you how it's done. This is the 5th step. The 6th step, sorry. Okay, and then click run. As you can see here, we have the composite, turn off the composite, and we have the MVI composite. We have that already. You don't have to display it anymore. Now this is the analysis result, the MVI INA. This one is the result of the analysis. As you can see here, it already mapped areas considered as mangroves, but since it also include areas of with low probability of mangroves, we have to mask those areas and create a new layer where it only shows the mangrove area for further analysis. So finally, you will get this area of mangrove. Okay. So with only 72 lines of code, you have been able to detect mangroves in your area using the mangrove vegetation index. Okay. Now, let's see. I'm going to stop sharing. Oh, I'm going to pause my share. And let's see if you guys happen to get any problems. Okay. Anyone succeeded? Let me see your result.
So,
yes,
Mr. Safran, can I share my screen?
Okay,
I don't know if
do.
it's okay. Share screen. So, does my screen?
Yes, it displayed correctly.
So I just want to ask a question before. So the before when I'm using the MVI, the threshold that I'm doing, it goes like I didn't find the gradation, and then when is on the step 6, is it? Yes, the step 6, the MSMVI and the MVIENA, it shows that the mangrove in this area. So the possibility as the Narja says before too, and as this MVI we can inspect as a mangrove, or we still should we still should check the threshold again or not? I mean, it's I don't know if it's right or not, like that's.
So I think your result is quite good, because I think this is still in Indonesia, right? So the threshold, yes, this threshold still, you can use the threshold as I made the threshold for Indonesia. So you don't need to play around with the threshold anymore, but maybe the other participant from other countries, maybe they require, or maybe they find new findings, and we can see Narjes already show us that she might have a different threshold.
Okay.
But this is already good.
Okay, so I just simplify that the Southeast Asia, especially there and Indonesia, that the project or the MVI is being used, we use the threshold like that, so we can maybe in the future when I use this threshold in area of Indonesia. Is it right, Mr. Safran?
Yes, correct.
Okay,
correct.
Thank you, Mr. Safran.
Okay, actually, we only have the next is only some extra steps. So extra step 1 is export.
Sorry, Mr. Safran, I think we still have several person want raise their hand, but unfortunately, I think this session will have to break at the 12 o'clock. But it depends on you.
Okay, I think I'm going to finish the extra step first, and then maybe before the beginning of the next session, in the next session, we can start with the question and answer first for MVI. What do you think, Mr. Bayu?
Yeah, it's up to you and also up to participant.
Okay, no problem.
Okay.
Yeah, because I see two people, I see 2% raise.
Okay, raise their hand. Okay. Okay, so the first extra step is exporting the data using this script. You don't have to do this, but it is sometimes it's important for you to have the data in your hard drive. And then the next step is the most important if you want to know the extent of the mangrove. So you can copy and paste this step 8 using this script. You have the var area image. So I'm going to explain a little about the script. So the first thing the script do is you declare a new image. In that image, each pixel, instead of MVI value,
we cannot see a screen.
Oh, sorry.
Yeah.
Sorry. I think. Oh, I haven't shared the screen yet. I'm sorry, everyone. Okay. So this is the extra step 1, extra step 2. So this is the extra step 1, export. I don't need to explain about the export. You just need to copy and paste the script and run it, just like the previous exercise. Now, what is important is in step 2, extra step 2. So you can calculate mangrove area by using the script. So what we do here is we create a new image from the MVI data, but instead of having the MVI value, we replace the MVI value with the pixel area. Okay, so each pixel will have a value of its area, and then afterwards we sum the values of the mangrove, and then you have the reduced region, and using the reducer sum, we sum all of the pixels to come up with a new variable, to come up with a new statistic, the number of the extent value of the mangrove in square meters. So if you please keep in mind that calculation in Google Earth Engine uses square meters instead of other metrics. So let's continue with the script. I will display it to you. Okay. I will just display the complete script and then check the console. There you go. You have the pixel, the extent of mangrove in the console tabs. Okay. These are the export, and this one is to calculate mangrove areas. Oh, I think in this script. Okay. Okay. Okay, you have the mangrove area extent by using the complete script, and you will also get the you will also get the mangrove area image in your cloud storage. Okay, so by doing this exercise, you will be able to map mangroves in your area and know the extent of the mangroves and have the shape file and have the image file for you to process again to lay out the map in Quantum GIS. Okay. Okay. Okay, I think I finished my extra talk. So we have 12:09. Maybe we have, I think, Patrice and Ruby. Do you have anything to ask?
Thank you, Mr. Safran. Can I share my screen?
Okay, please do.
Okay. Can you see?
Okay, you have.
I have 2 errors at the end of the script, but I cannot identify what's the problem.
Oh yeah. So this is because it's an incomplete incomplete script. So you still need to copy and paste the extra. I will give you in the chat. Okay, that one. So replace the step 8 to the bottom of the screen with the snippet I've already given you in the chat box. Okay, I think we should be okay after this.
Okay, let me stop my sharing.
Okay. Okay. Patrice, you still have a problem?
Well, I won't. Can you see my screen?
Yes, yes, clear.
Okay, so I just wanted to confirm that this was what I should be seeing.
Yes, right. Use the mask MVI, not the MVI INA. Yes, mask MVI. So that's your mangrove area.
Yes.
Mangrove area. Yes, I think you are correct. Please check the console tab. Please check the console tab. No, the console tab. The console tab. Please run the script again, one more time. The script run one more time, and check the value of the pixel. Now you have the value of the pixel.
Okay.
Okay. Okay, Patrice, congratulations.
Thank you.
Okay, congratulations, everyone. I think you guys have already mastered mangrove mapping using MVI. So we're going to take a break. Afterwards, we're going to use machine learning to detect mangrove areas. Okay? Congratulations, everyone. You guys are super.
Yeah, thank you, Pak Safran.
Okay. We'll see you in 45 minutes, Mr. Bayu.
45 or 1 hour.
Okay.
Yeah.
45 minutes. Okay.
Just try to following the schedule.
Okay.
Yeah. Okay.
Nice.
See you later.
See you later.
Yeah, see you.